Good morning, everyone. Welcome to this workshop that we have organized to celebrate the 30th anniversary of the CNB. Especially, I would like to welcome and thank the invited speakers, one for each department, as they are former members of the Institute and come back to tell us about their scientific activities and to remember their time at the CNB. The CNB is today one of the biggest institutes of the CSIC and probably the most interdisciplinary one. Although originally this interdisciplinary focus was considered a disadvantage to compete for funds and to create a common strategic plan, today the real situation is that our multiple focus on science is perhaps the most important advantage when we compare with the CNB with other institutes. In any case, this is the way that we choose to answer scientific and social problems, as well as the way to improve our projects on health, agriculture, and environment. As an example, I want to refer to our work during the recent pandemic caused by SARS-CoV-2. We have developed an amazing amount of work during the last 2 years that can be summarized with these small indicators. 56 papers published, 14 patents developed, 24 people hired, a kit of seroprevalence licensed to the WHO, and 2 vaccines prototypes very close to the clinic. During these 30 years, we have developed an enormous effort to incorporate new technologies and established several technical units to help scientists. As an example, a couple of words to indicate that, as you know, we have completed a bioimaging platform that has been created to allow researchers to evaluate from organisms, organoids, cells, molecules to atoms. The platform also includes 2 units to extract the data from the images to allow quantitation of biological processes. The next step now is to incorporate these technologies to our current activity. Our new strategic plan is now focused in the incorporation of young scientists to renew our staff, in improving the internal collaboration and the use of the available technology, and in creating links with other national and international institutes to increase the internalization of the CNB. For that, we are going to focus in 4 areas: anti-infective therapies, new drugs and immune-based strategies, advanced biotechnology of the microbiome, sociology of the cell, and artificial intelligence and automatism in biology. But well, now it's better to listen science. It's time to enjoy the science and the talk of our invited speakers. And therefore, I think that we are going to start directly the workshop. Then the first speakers comes from the Department of Molecular and Cell Biology. And therefore, Paco Rodriguez has now the microphone.
Okay, good morning, everyone. And thank you, Mario, for not only you, but all the rest of the people at the CNB, especially Susana and Ricardo, for organizing this very nice and interesting anniversary meeting. Well, I have to say that I'm very, very proud to introduce our first speaker today, who is going to be Antonio Maravera. Antonio, I think, is a very, very good example of the CNB's scientific seeding capacity. Antonio first joined our group at the Molecular and Cellular Department some 24 years ago, in 1998. And at the time, he was a very keen and very bright and enthusiastic graduate student. And time has passed, and I can say now that he is a very bright and probably even more enthusiastic senior scientist. Well, when Antonio joined the lab, I shared with Eleuterio Lombardo, Lele, for people at the department, who was a young person then, the direction of Antonio's thesis that was centered on the characterization of the transcription replication complex formed by 2 proteins of infectious bursal disease virus. He worked during 4 years, and I think he was, at the end, he was a little bit fed up of virology, and he decided to switch and to make a big change in his career. From virology, he decided to move into cancer. Probably it was a very wise movement. And then he left Madrid and he joined the Scribble Institute of Molecular Medicine at NYU, where he was working in cancer research for about 3 years. After that period, he decided to come back to Madrid to join Manolo Serrano's lab at the CNIO. First, he got Juan de la Cierva, and then a Miguel Servet contract. And after 9 years, almost 9 years, of very, very intense and successful research and publishing a number of very, very seminal papers, he decided that it was about time to get his own research going on. And then fairly soon he got a permanent position at the INSERM in France. And he started his work as an independent researcher at the Institute of Cancer Research in Montpellier, where he has his own group. And it is a fantastic group, I would say. I visited him a few years ago, and I was really impressed. His work now, I think he will explain it in more detail, but I find it extremely exciting. It is a combination, which I find very interesting, of basic and translational research with a primary focus on establishing rational combination therapies for lung cancer. Well, thank you, Antonio, for joining this anniversary workshop, and you have now the word. Okay, thank you.
Thanks a lot, Paco. It's always a massive pleasure to come back home. It's only a pity that I cannot present downstairs in the big seminar room because I'm physically now at CNB, just in the office in front of Paco. So thanks, Fernando, for sharing your office with me today. I'm going to share you the presentation. You can see it, right? As Paco said, I started here in 24 years ago, and I came back today in the 740 bus, and a lot of thoughts and memories came to my mind. That was a beautiful period of my life. And actually, I remember perfectly the first day that I came here to the CNB. It was thanks to my professor that I did have in the university, Antonio Maravera, because I studied in Alcalá, and we don't have really good connections, at least at the time, with the research institutes. It was a bit, so he told me, no, you go there and you ask for some people, he gave me some names, and I came with a CV, and I've been treated super nicely. I remember that Mariano Esteban hosted me to talk a little bit, and he told me, you know, there are no space now in my lab, but I have a colleague, Paco Rodriguez, is looking for. Paco was at the time doing some holidays. When I came back, I was totally burned because it was red. It was kind of weird, and it was a fantastic moment. He basically told me that there was a position, but he offered to someone else, and he's super fair and a fantastic person and honest. And basically, I say, okay, I understand, but I really would like to do it, blah, blah, blah. And then 1 week later or something, he called me and said, you know what? The person that I offered, he said no. And then I came back and we discussed a lot. And I remember myself, all of a sudden, I realized what it was when I was on the subway coming back to my home, and I was crying like a baby, because science is a lot for me. I'm extremely enthusiastic with science. I really love our job. I think that we are very happy with this. What can I say? Anyway, so as Paco said, I'm now in the Montpellier in the Cancer Research Institute. And I'm going to show you, it's a kind of difficult exercise for me. How is the whole, my whole career, let's say. And I will focus a bit first with the PhD that I started with Paco. And basically it was regarding the role of the polymerase BP1 and a scaffolding protein called BP3. And we passed really good times doing fantastic research. And I learned a lot of molecular and cellular biology. I became a real expert. And basically, we published very well. It was very successful. But not only that, actually, Paco has a lot of good themes. And one of them is that actually he's super open mind and keen to let us move. And I was eager to do it. So I did 2 stages, one in the University of Turku with Olivainho, and then in the Scripps with Gary Bokoc. And the thing is that for the second one, it was thanks to Lele, that as Paco said, he co-signed and as you see, he was last author in this particular paper because Paco is super fair and actually it was real. So Lele did a lot of work and it's normal that he's there. And we were working super hard in San Diego. So he was there in San Diego and helped me to get started. And we worked a lot, particularly to find the good outfit for this party that we did like 1 week later. And at the time, even some of the colleagues that I have here that they are PIs, like Pablo Gastavinza, at the time they called me Jim Carrey. You know why now? And Paco obviously was aware of all this, and we was a bit worried. Sometimes I think that he was about to take a strong decision, but thanks God he didn't do it. And basically coming back to the first stage, actually. With Oli, he was an expert in immunology in chickens, and I found this quite interesting. I really enjoy, but Banksy showed me the way. So actually, I was ready to move to study other complex organisms, meaning mice. And the only thing that it was lacking is to know where to do it. And Paco again was very important because he was always telling me super nice things about the capital of the world, that is New York. And I passed years there, that it was fantastic. That's the view of the Empire State Building that I was almost having from my house, a very similar one. And there in the lab of Juan Lafayette, I started to study T-cell development. And during the T-cell development, I will go very fast, but basically you know that you have the double negative 2, 3, 4, and then you become double positive. So it was known at the time that the Notch pathway, it played a major role in this particular part, but it was not known what happened after double positive. So what is Notch? Well, it's a pathway that it gave me a lot during my career, and I have to tell you a little bit. So I'm kind of super geek scientist, and I found fantastic this paper from Morgan, that is genetics that made a lot for genetics using Drosophila as a model, and this is why it's called Notch. It's just little notching here at the end of the wing, and you could say, well, what a phenotype! This is really small. But the thing is that actually he discovered that it was in the X chromosome, and this is why you only see the phenotype in females. But on the contrary, when it's in homozygosity, it's lethal, and of course it's lethal also in males. So I don't want to bother you too much, but you need to know only that it's a very peculiar pathway because you have one cell that has the ligand on the membrane, and another cell that is the receiving signal cell has the notch, that is the receptor itself, also a transmembrane protein. And then upon interaction, you will release notch ICD at the end after the cleavage by the gamma secretase complex that you will see is important because we target here with inhibitors. So why I found so interesting, as you see, this is a phylogenetic study performed with the pathway, and very early, almost at the very beginning, when 2 cells talk to each other, or they needed to do it to make an organism in the animal kingdom, Notch was already there. And this is telling you how important it is. This is a super simplified version. So once again, when the receptor it is processed, it is proteolytically cleft by the gamma secretase, you release the notch ICD, interacts with a DNA binding partner that is a PPJ, and you get several readouts, as for instance HES1, that is a target gene downstream. So now I can come back to this T cell development, and what we did, it was a genetic trick using a presenilin-1 fluxed animal, and we eliminate using a transgene of CD4-3, meaning that the recombinase will work only at this DP stage. What you see is that actually the typical bird in the thymus, it doesn't move at all. So basically the notch pathway was not required. So we published this long ago. And then the other part is what Paco was saying, that is true. So basically, I was involved also in acute T-cell lymphoblastic leukemia, where notch, it is extremely important. So what we did, it was to eliminate this in a transgenic mouse model that it does express one of the ligands that is called DLL4 on the membrane. And this is one result that it changed my life. And this is what I'm putting here. So basically, this is the survival curve of these animals that are called TG8, that they develop acute lymphoblastic leukemia, and in 120 days, all of them are dead. If you eliminate only one allele of presenilin, you already see an effect. And when you eliminate both, the acute lymphoblastic leukemia, it gets cured, because this is an escaper. Actually, notch was on in this particular one. So we published this back quite late, and I was following Xion with Juan, and this is why he shared with me the corresponding that helped me a lot at the time to build my lab. Juan was super nice with me always. And then something very weird happened, and is that the animals, these animals that they were cured, they also started to get the lymph nodes very big. And it was not because of the leukemia, but something lymphoproliferative, that it was not cancer. And one day I remember that I was getting out of the institute there, and all of a sudden I saw it. The thing is that once that the T cell arrived to DP in this transgenic, they do express DLL4 on the membrane. And this, in the periphery, is going to show to the progenitors. And what we did have, it was T cell development in the spleen. So basically here you see the DN2 and DN3 that are these phases, but this is happening in the thymus, but here is in the spleen. The normal spleen, of course, it doesn't have anything. So what we did, it was to remove the spleen of the mice, and they get cured. So we cure first genetically, and then secondly with a splenectomy. So we published this very recently, because it was with the help of Michael, that is a brilliant postdoc that is in my lab. And he helped us to finish. And basically, as you see, you need to be patient in science. I left the lab in 2005. And this is 2021. I think that this is very important for the young generations. You have to keep faith in what you are doing and keep pushing till the end. Then I came to this other area. This is a gut that I found in one museum in Oaxaca, in Mexico. So it's a Zapotec gut that it has several thousand years, and I find very nice for what I study when I joined the lab of Manolo Serrano. I remember the day that I met him finally, because it was first by exchange, and he came to a meeting in Cold Spring Harbor, and I remember him. I'm seeing it. He drew the survival curves that he was having with some animals with a bit of more P53, and I saw it. This is outstanding. I got a gut feeling that told me this is going to be a great story, and actually it was. Indeed, it was. And I did this job with Ander, with my friend. Maybe you remember him. He was here with Manuel in the fourth floor. He was doing the PhD. We are super nice friends, and we share the co-first in this study that helped me a lot for several things. And at the end, actually, as Paco said, I obtained a Miguel Servet, and this is where what I did. It was to brought all the models of the notch pathway that I was using in acute lymphoblastic leukemia to study in solid tumors. And what we did in this first, it was to inhibit either the presenilin or another animal that we obtained from the lab of Onjo in Japan, of RBPJ, that are fluxed also, and how we eliminate, it was using an adenocrete and injecting into the urinary bladder, and then we gave some carcinogens specifically for the bladder, and what you see is that both, they do develop the carcinomas, well, they die of the urinary carcinomas way faster than the wild type. So we published this story very well. And I have to say that Manuel was extremely nice with me, and we say the corresponding authority of this paper. He was fair because the notes I brought it and the animals, but he was super nice. And this, of course, helped me a lot during the establishing my lab. Then probably the story that most important that I published, because it changed my life in the way of where I would make my lab, my laboratory. And it was with this, I found this mouse in Paris when I went to get my permanent position. I was super lucky. I got it at first, and this is very rare. And basically what you see is that these guys are working, because our mice are working a lot, the poor little things, and this is smoking. So this is already telling you what is going to happen, because it's a lung cancer, indeed. And I will explain you a bit better this paper, but first I'm going to explain you why I decided to work in lung, because I published this, but we published in bladder, we published in thyroid cancer, several different models in the lab of Manuel that is extremely eclectic. And then I decided to focus. I remember it was in an ASEICA meeting, that is the National Cancer, Spanish National Cancer meeting, and I saw a clinician showing survival curves of patients, and just 1 month of extension was like, wow, this is fantastic. And I was so depressed, and I said, we have to do something here. And this is why I decided to basically dedicate my life to lung cancer. So just to let you know, these are the 10 causes of death back in 2000, and there is only one cancer among the 10, that is lung cancer. 15 years later, the situation didn't get any better. Actually, it's something it gets worse. There are several reasons for that. And here I like a lot, even if it's a bit outdated, it's very nice because we see how much it increases the survival of patients during 40 years of research. And basically what you see is that in some cases it's outstanding, but we go down, and then we have lung and pancreas. Everybody knows that are the deadly, the worst in terms of prognosis. And basically the difference is in here, that is lung is very prevalent. So this is why basically this is the situation. In France, this is where I'm working, and it's not the most prevalent, but still is the leading cause of death. So just to let you know a bit of knowledge, based in the histopathology, what we have is that our tumors that are non-small, meaning that they are big, and then those that are small, and then this is subdivided in another 3. This is just to explain you that in my lab, that is small, we only work in lung adenocarcinoma to get focus. One idea that I would like that you would remember today with this, that would be very nice, basically is that what we have is the in the normal tissue, it is going to happen a hyperactivation of oncogenes and then the inactivation of a tumor suppressor. And the thing is that at the end you get a malignant transformation, and here is what is interesting, because they become addicted to the oncogene of origin. And this has been exploited in lung adenocarcinoma very nicely. So for instance, we have EGFR mutated patients that were treated first with first generation TKIs, gefitinib or erlotinib, but all the patients became resistant, all. And more than half, it was because of this so-called gatekeeper mutation. Then we have now osimertinib. Then they do develop also another resistance, and even more, they can get resisted, they can relapse also by amplification of MET. That is another oncogene in itself, or you can have the AML4ALK. But the thing is that in all of the cases, we have small TKIs, tyrosine kinase inhibitors, that they are very specific and it works very well. But again, they always develop resistance. And then we have KRAS that we didn't have targeted therapy at the time. Now we have sotorasib, only for the KRAS G12C, particular subtype. And this is just one part of the story. because the other is that in people trying to believe that that cancer, they have a nice life. No, they are facing stress all the time. And actually, there are plenty of different types of stresses, like oxidative stress, etc., etc. But the interesting part is that all these pathways that are used to stop this, normally they are not related to the oncogene directly, but some other pathways that are hijacked by the oncogene, and is what it's called a non-oncogene addiction. And in the lab, we use the Notch pathway as an example of this non-oncogene addiction. So once again, the Notch pathway, don't want to bother it too much, but please remember that the gamma secretase complex that is here, we have good inhibitors. And this is only an example of some clinical trials that are making now with gamma secretase inhibitors. So once again, now I can explain you a bit better. The first proof of concept of the role of Notch, it was here. So we, using the RBPK that I showed you before, we did also with the presenelin, but just by lack of time, I showed you only this. So we demonstrate that the carcinomas requires the Notch pathway. So in other words, if you don't have the Notch pathway, the cancer do not happen. You get benign tumors, but you don't get cancer. And to get it more translational, we treat it with a gamma secretase inhibitor that is basically going to inhibit here. And what you see is that following the tumors by PET, that is exactly what we use in the clinic for patients, we were able to demonstrate that even with the monotherapy alone, the KRAS tumors that are very, very, very strong, they were arrested. So this is how I started my lab, let's say, with this idea of what it could be the role of Notch in all those other different pathways, oncogenes, sorry, and with the difference that in this case, we can combine at the same time the non-oncogene addiction and the oncogene addiction using these small TKIs. And I've been, during the last years, acquiring or developing new mouse models, the genetic engineered mouse models to mimic all these diseases. And then we don't forget KRAS for the chemotherapy that are with carboplatin or cisplatin, and then now with sotorasib. So I will explain you just very fast at the models that we have. So the first one is the EML4ALK, and we use it using this CRISPR-Cas9 system, that it works beautiful. So basically, you introduce an adenovirus into the lung of a wild-type animal, and Cas9 is expressed, and also sgRNAs that are going to recognize EML4 and ALK and are going to make de novo the EML4 translocation. And once again, it works beautiful. How we do that? So we put the mice like you see with a strong light, and here is just this white thing is the entrance of the trachea. So we intubate the mice. We put with a pipette the amount of virus that we want, and then with air. We make a bit of air, and here we have some blue just to show you how basically it looks. This mimics perfectly the disease. That means that we need to follow the tumors with computed tomography, and that was thanks to the help of colleagues there in the IRCM in Montpellier, because this is extremely complicated to do with the synchronization of the breathing. We presented this to Roche, that they were very interested because they have a TKI called alectinib. And first thing we did, it was to demonstrate that that was a good preclinical mouse model, and indeed it is, because it behaves as it should. So they do respond very well to alectinib, these tumors, followed by computed tomography. And we got antibodies from Genentech and a lot of money. And this is what I want to stop for this particular one. But we have the story very well advanced. And then the story, probably the best example that we have done in the last years in the lab. So is with this mouse model that basically is a classic teton with an EGFR that has the activated mutation and the gatekeeper mutation, meaning that they will develop cancers that are resistant straight to first generation TKIs. So then we express RTTA with another specific promoter from the lung, and then still it doesn't work. Then we have to add the doxycycline that we put it in the food. Extremely expensive, but super nice. It works beautiful. And then only now we have the expression only in the lung of the EGFR. And this, my colleague KK1, published years ago that it develops carcinomas. And indeed, it worked beautiful. So this is the experiment we did. The first, we gave 8 weeks of doxycycline to these mice. They start to develop tumors. And then either we gave only gefitinib, first generation TKI EGFR, then we gave the notch inhibitor, or we combine. Here what you see is that the vehicle, it has a lot of tumors. When we give the gamma secretase inhibitor, we did have a nice effect. On the contrary, when we give gefitinib, obviously, there was no effect. There was nothing like giving PBS. And when we combine, it was outstanding. So basically here we have the quantification of the tumor burden. And once again, what you see is that the combination somehow was doing to work back the gefitinib. That was kind of alchemy. So it was super weird. And actually, we confirmed this also with the adenocarcinomas. So the only treatment that reduces the number of carcinomas, it was the combination, and that was really outstanding. So that was outstanding. And the thing is that it was so weird, so unexpected, that we decided to use a second preclinical model to prove that that was true. And then for that, we have a extremely good collaboration with Liz Pasares and Irene Ferrer in CNIO and Hospital 12 de Octubre. And basically, we use patient-derived xenografts. For those that they don't know, basically, you get from the tumor of the patient, you can grow in immunocompromised mice, and then you can expand. And we were lucky enough that they did have one PDX that it was having exactly the same mutations that our genetic engineer mouse model. So we replicate the experiment with subcutaneous growing of this PDX, and basically the result you see here is beautiful. So the gefitinib basically did nothing. The GSI was having an effect, and the combination was very strong. So how this works, I have no time to tell you everything, but let me simply put this under normal condition. Resistant cell line that is treated with the EGFR TKI is going to increase levels of phospho-STAT 3, and then Notch ICD is going to occupy the promoter of HES1, and we have expression of HES1, and that's it. On the contrary, when we co-treat with the gamma secretase inhibitor, Notch is not able to, Notch ICD is not produced, and this phospho-STAT 3, it binds and inhibits HES1 expression, and this is enough to induce the resensitization. And we published this very nicely in 2020. This paper was a pain to publish. It took me like 2 years of suffering. I cannot tell you how much, because the story, I knew it was good, but still we were not able to pass the editorial process, whatever. And well, anyway, just to let you know again, for the young generations, if you have to trust in yourself, you have to trust in your ideas, you have to keep pushing till the end. And then I'm going to tell you a very fast another story, because Michael needed to get more stories to publish. So we decided to do a quick and dirty, well, not quick and dirty, let's say quick and fast, very fast paper to show something, to get another paper to get the permanent position. So as I told you, today the patients EGFR mutated in lung adenocarcinoma treated with osimertinib, and more than 20%, they do relapse to osimertinib because they have an amplification of MET. So we decided to cross our EGFR mouse model that I just showed you with another oncogene with MET, that they will overexpress MET. And basically, it works beautiful. So here you see that when you treat the EGFR only mice with osimertinib, they do respond very well. This is expected. That was developed for that. On the contrary, you see there was no response when we put MET, mimicking what is happening in 20% of patients. And as it happens in the patients, only when we combine the crizotinib, that is basically another TKI for MET, well, it's a pan TKI, but it inhibits very efficiently, and it's given in the clinic to patients that are MET amplified. Only the combination of crizotinib and osimertinib work. And I'm putting all this because we have a very nice interaction with the clinicians in our local cancer hospital called ICM, and we have several of them in our lab. And in the lab meeting, Camille, that is fantastic, she was seeing the data and all of a sudden said, oh, but I have a patient that we are treating with this and is responding very well. And I said, let's put it. And the last panel that we have in this paper, it is a patient that is showing a nice effect when combined. A EGFR patient that relapsed to osimertinib by MET amplification. So we published this is the fastest paper I ever published in my life, I have to say. And the good news is that Michael got a permanent position, and this is probably the best thing that happened in my lab in the last years. Then Michael also developed a mouse model to make lung adenocarcinomas only with MET, mimicking the MET amplification alone as a driver. and it works very well. So this is just to show you a carcinoma. This is easy to see because it's surrounded by the bronchia, so it's invading. And I have no time to tell you this story today, I'm sorry. And actually, it's probably the most exciting study that we have done in the lab. I'm extremely, extremely excited with the data. It's beautiful. And actually, I've been invited as guest speaker in the next ASECA. So if you guys are going to be in Santiago, please come. I guarantee that you will enjoy a lot. And basically, with this, I'm going to tell you a little bit about our the last thing I want to tell you today. So we show something very interesting in cells that we see an increase in notch activity, notch ICD levels upon carboplatin treatment in cells. And this is that was the PhD thesis of Sarah that now she left. And basically, the models that we use, it was because Irene, that I told you before, is in the lab of Luis Pasares. She came for some months to my lab. She's fantastic. And basically, she wrote the patient-derived xenografts that she developed. I'm going to show you very fast these 4 models that are all KRAS P53 mutated, extremely aggressive. And what we did, it was to combine the carboplatin treatment with a gamma secretase inhibitor and to inhibit the notch pathway once again. And basically what you see is that in the 4 models, we always observe a better therapeutic response when we combine. That was pretty exciting. And we decided to do something very, very complicated with an IZG, that is basically a super technicians with hands that are pure gold. And basically, she treat for very long period, more than a year, was giving treatment, then let the tumors regrow, then give, then regrowth, to try to generate resistance in vivo in the PDX as we do with the cells in vitro. And we failed. And this is why I wanted to tell you, because not everything that we do in the lab, it works. But this is bullshit, obviously, it's not the case. And I like to tell you the whole thing. But I proposed to analyze something way more complicated, that it was to put back the tumors into the lung. So doing a thoracic surgery in the mice. And that was really challenging, and she's brave and she managed. So first you have to intubate to put a respiration machine, because in the moment that you touch the lungs, they collapse. So like in patients, you need to intubate. And this intubation is way more complicated than the adenopathy. And it took us really, really a lot of time to put it to work. But at the end it did. And now just with the nude, to show how it is. So you put a little piece that is like this, so it's, for the mouse is not small, and you put it between 2 lobes, and then mice are extremely strong, they are amazing, I love them, what can I say? And 1 month later, what you see is this. So this is the xenograph pushing out from the inside. So this is not yet cut. So here you have the ribs, and then we cut the ribs, we put the whole lung out, and this is how it looks. And when we have a look in the distal lobes, not in those that are already eaten by the tumor, what you see is that we have local metastasis. That is extremely interesting. You need to be a pathologist. As I told you, it's non-small, so meaning they are big. It's easy to see how they are going.
Sorry, Antonio. I have to say that you are really getting too close to the end.
Okay? Yeah, it's the end, Paco.
Okay, fine.
It is the end.
Fine. Thank you.
So I just want to say that this model, it brought us a fantastic European grant. So this is why I wanted to put it, that the models, they brought the attention of the investors. And to finish, this is the overview of what I wanted to tell you, that what we have and what we do in the lab. And basically with this, and the 2 stories that I told you today very fast. And basically with this, I want to finish. I want to thank all the people that worked during the last years, the money givers, all of you for your attention. And I want to have, again, some words for Paco and Lele. I don't know what I am, but whatever I am is thanks to you. So thanks.
Well, thank you, Antonio. As I told you before, Antonio is a very, very enthusiastic and energetic scientist. Thank you, Antonio. Antonio, you can stop sharing your screen.
Yes.
I would like you to have any questions for Antonio's presentation. Okay. I'm waiting for the questions that are coming through YouTube. In the meantime, okay. Susanna is telling me that there are no questions so far.
No
but
problem.
Just
everything was so clear.
I have a good one for you that is philosophical. Well, Luis Montoliu is saying thanks for your talk. But my question is that did you ever regret leaving virology and switching to cancer, especially after the recent pandemic? Probably you've been crazy about working on that. Positive.
Well, actually, I only say things in a way that is historically like nice, but actually this European grant, a massive one that we just obtained, came in the very good moment because we were about to get absolutely anything. For a couple of years, I was seeing like people was publishing in super good journals things with a couple of immunoprecipitations of the SARS-CoV-2, and basically, of course, I got a bit upset, but no. Actually, what can I say? I enjoy every single step that I have done in my career, and I love the lung cancer now, as I did love morphogenesis of IBDB. And if I would be working in I don't know what, I would love it because I get enough.
I have a question here for you from Eva Gonzalez.
Yes.
She said, increasing notch after chemo only with platinum.
Yes.
Also with other chemos.
Yeah. This is something that actually is, well, Eva, I didn't say anything. Hi, Eva. Basically, it is, we do have not only the hypothesis of the mechanism, but actually we know exactly how it works. So the DNA damage is going to induce the activation, well, it's through ATM, it increases levels of MDM2 in a P53 independent manner. So we have found a new axis that is ATM, MDM2 notch ICD. So what it does is to ubiquitinate notch ICD, and it stabilizes notch ICD. So it does not induce its degradation, but the opposite. So it gets stabilized. And this is what we are about to submit now. We are just waiting to get some data from patients. Thanks to Luis Montuenga in the CIMA, that we have a super good relationship also. And this is what we are waiting to stain in the TMAs for MDM2 and HES1. Thanks, Eva.
When if it's finished, thank you, Antonio, for this amazing and enthusiastic talk. Now is the time for the Department of Macromolecular Structure. Please, Carmen, go ahead.
Thank you, Mario. Good morning, everyone. So today, it's a great pleasure and an honor to introduce Shores Jerez in this 30th CNB anniversary. Shores graduated in chemistry in Utrecht University in the Netherlands, and then he did his PhD in chemistry there also, where he was working on the development of methods to process protein crystallography data. In 2003, by a combination of fortunate personal and scientific interests, he came to Madrid and joined Jose Maria Carazo's group here at CNB, where he worked as a postdoc for a lot of time, 7 years, until 2010, when he moved to the MRC Laboratory of Molecular Biology in Cambridge to start his own group. There, Shores has been working on the development of software tools for the analysis of cryo-electron microscopy images. It was very fortunate that he decided during his time in Madrid to transition from crystallography to cryo-electron microscopy, because he now has one of the most widely used programs for cryo-electron microscopy. And also, he also arrived at the MRC in a sweet moment when the first direct electron detectors were starting to work. And you have already heard about the cryo-EM resolution revolution. Shores participated in one of the first studies showing that with images from direct electron detectors, you could get very high resolutions with a reasonable number of data, a reasonable amount.
Carmen, te has perdido.
Of data. No? They have actually reached the true actual. Sorry. Well, hopefully I'm back. Then Shores decided to start applying these tools to a very relevant subject, such as the structure of the filaments found in patients with Alzheimer's disease or other neuropathologies. And this has also opened a very new field and now a wide field in structural biology. I'm not going to say anything about Shores' publications, just want to say that he has over 25,000 citations. And he was elected a member of the European Molecular Biology Organization, EMBO, in 2017. He has received many awards. He's a fellow of the United Kingdom Academy of Medical Sciences, a foreign member of the Netherlands Academy of Sciences. Recently, he received an award called the Ivano Bertini Instruct Eric Award. And for the structural biologists in the audience, I just want to mention that the previous person to receive this award was Wolfgang Baumeister. So you can have an idea that this is not any award, that this is an award that important people receive. Finally, I just want to highlight that Shores is an advocate of open science, distributing his software widely to the community. And in this sense, we have also to thank Shores for his involvement in giving access to the Spanish cryo-EM community to our first access to microscopes with direct electron detectors back in a time when we didn't have this kind of equipment in Spain. So thank you, Shores, welcome, and the floor is yours.
Thank you very much, Carmen. Can you see my slides?
Yes,
yes,
we can.
I fondly remember the times of the early revolution when you guys would come over to the LMB and collect your data. Those were good reunions. So I'd like to speak today about developments in cryo-electron microscopy, image processing, and I'll tell you both about work done at the CNB as well as at the LMB. I thought perhaps for those not so familiar with cryo-EM, I'd give a few introductory slides. So here you see an image that was taken now almost 10 years ago by a postdoc from the Ramakrishnan lab here at the LMB, somebody who many of you may be familiar with as well, Israel Fernandez, who was before at the CIB in Madrid. So these are E. coli ribosome particles, and the purified solution of ribosomes from Israel was pipetted by a postdoc in my lab, Xiaoshen Bai, and deposited on a small metallic grid about 3 millimeters across in diameter with a pipette, and then using more or less filter paper, the excess liquid was blotted off, that created a very thin layer of buffer solution in which these ribosomes were suspended, which was then rapidly frozen in liquid ethane, which leads to such a rapid freezing that the water does not have time to crystallize, and you get this amorphous form of solid water or vitreous water. And in that, you can then look look at the ribosomes in a transmission electron microscope, and that's the type of image that you see here. It was collected on one of the prototypes of the Falcon 2 direct electron detector, which Carmen already referred to, which became available at the LNB early 2012, I think. So in the image, you can see each of these little black dots is a ribosome particle. And because the sample was frozen when these were all tumbling around in random orientations in this thin layer of water, each of the two-dimensional projection views that you see here is hopefully an image of the ribosome in a different orientation. And these can then all be combined together in a three-dimensional reconstruction to have a look at the actual three-dimensional scattering potential of the ribosome particles. How that works, I think, is best explained through the projection slice theorem. And it works as follows. We have a three-dimensional structure, and this is actually gamma secretase, which was mentioned by the previous speaker, which we did the structure of together with Yigong Shi in 2014 and 2015. But if you have a three-dimensional object, you can discretize it on a volumetric Cartesian grid, and then you could calculate in the computer a three-dimensional Fourier transform. That's just a mathematical operation. It's a different way of expressing the exact same information in Fourier space. And you could go forwards and backwards just using your computer. Now, what happens inside the microscope is that along, for example, the path of the red arrows here, you get a summation of scattering density, more or less. What you get in the image is a two-dimensional projection image, a so-called X-ray integral is what the mathematicians call it. And now for this two-dimensional image, you have the same. It's collected on a pixelized detector, and you can then calculate the discrete two-dimensional Fourier transform of the two-dimensional real space image. Now, the projection slice theorem now says that this two-dimensional Fourier transform here is a central slice through the three-dimensional Fourier transform of the original 3D object. And it goes through the origin of the Fourier transform, and its orientation is perpendicular to the projection direction that the 2D projection was made of. This means if you would have many two-dimensional Fourier transform of many of these ribosomes, all in different orientations, they would all come together. If you would know all the relative orientations, they all come together, filling up as a kind of three-dimensional spider web, the three-dimensional Fourier transform. And then all you need to do is an inverse transform to get back to the three-dimensional structure of the ribosome. So how we do that is through a method called projection matching. And basically we start from some low resolution estimate of how the structure might look like. And modern algorithms don't need that to be any more informative than just a more or less a spherical ball of density without any information. But I'll start with a bit more informative one here just for illustrative purposes. In the computer, you then make reference projection library, which is just we simulate what the 2D projections of this three-dimensional structure would look like in all possible 3D orientations. Then for each of the noisy images that we've recorded and cropped out from that large image that I showed you in the beginning, we then use the computer to compare it with all the reference projections and see which one fits best. And you can formulate multiple criteria on what best would look like in a mathematical sense. And then knowing now the best orientation for each of these particles, you can then put all these particles back and do this in the 3D Fourier transform and do the inverse transform, getting back this three-dimensional reconstruction of the particles. Now, because the orientations assigned were based on a relatively low resolution estimate of the initial signal, the orientations won't be perfect. But the reconstruction that is made will mathematically be guaranteed to be better than the one that you started off with. And that means you can then iterate over the whole process. So you can take this better structure, do the whole thing again, make better reference projections, get better orientational assignments, get better reconstructions, until hopefully, and this is not guaranteed, the whole process converges onto the correct structure of the three-dimensional object that you're interested in. Now, as Carmen mentioned, I was at the CNB from 2003 to 2010, where I was a postdoc in the group of Jose Maria Carazo. There, we started thinking about this problem. And if I think back on this time, I think the main contributions of my work at the CNB was the introduction of a concept that was very familiar for me from coming from X-ray crystallography before and methods development there, which is the concept of marginalization, or as it has become known in both the X-ray and the cryo-EM field, is maximum likelihood. And this is a concept where there is all these parameters that we're not really interested in. All we're really interested in is the three-dimensional reconstruction of the object of interest. And all these relative orientations are so-called hidden variables, or they're part of an incomplete mathematical problem. And there is a statistical way of dealing with these type of variables, which is marginalization. And basically, it depends on a statistical data model, which then used to calculate probability weighted assignments of all the possible orientations. So rather than just assigning the very best orientations, each particle will contribute to all orientations, but these are weighted according to probabilities that are calculated based on this statistical model of the data. We did that for two-dimensional classification of images and later for three-dimensional classification of images. First instance in real space, and then later we also made an expression of this probability in Fourier space, which, as I hope to convince you from my first few slides in the Fourier slice theorem, is actually a more natural space to do this in. So together with those more fundamental methodological advances, Jose Maria and I published together 23 other papers. So overall, I think my stay at the CNB was very productive. Now, as an example of what you could do with this three-dimensional classification, the first application we had was a collaboration with Joachim Frank, to which Jose Maria, of course, had very close ties, because that's where he was before coming to the CNB. So Jose Maria had done this reconstruction of, Joachim had done this reconstruction of E. coli ribosome particles. And they had combined 91,000 of these individual images of the ribosomes and calculated, this is prior to the resolution revolution. So this is data collected on photographic film. They calculated this 10 angstrom reconstruction, which showed some problems because there was relatively good density for the three transfer RNA molecules here, but density for the elongation factor G, which was supposed to be present in the sample, was rather weak, and density for the 30S subunit appeared blurred compared to density of the large subunit. Now, using 3D maximum likelihood-based classification, we then did a multi-reference refinement. So we did this projection matching against 4 references at the same time, using these probability-weighted assignments, not only for the orientations, but also for the class assignments. So each of the 90,000 particles would contribute to all 4 of the classes, but with different probability weights. And if you go through this iterative process 25 times, which back then on the computers available in, this was around 2006, 2007, it took half a year of computation on computers. And for that reason, we then also, again, with good help from Jose Maria, got access to the Barcelona Supercomputing Center, where we do these computations massively in parallel, being able to finish them in just a few days. The results were 4 different structures, 3 of which were more or less the same version of a 70S ribosome with the 3 tRNA molecules bound, but the 4th structure was notably different. Only 1 tRNA molecule was bound, and EFG was bound to this one. And because these were only 16,000 particles out of the 91,000 particles, this kind of explained why the initial structure had relatively weak density for the EFG. Also, the orientation of the small subunit, the yellow one, compared to the large subunit, the blue one, was different in the 4th class, kind of explaining the blur density that they observed in the first structure. So this was the first example of being able to separate out structural heterogeneity, distinct 3-dimensional structural states from cryo-EM data sets without knowing a priori what was the difference between the different structures. So Then in 2010, I moved to the LMB, and again, the CNB played a role in this. In 2009, the students at the CNB decided to invite Richard Henderson to come and give a talk. And based on discussions that I had with Richard there, and Richard asking people at the CNB who they would turn to if they would have problems with image processing, and I'm grateful many of you actually mentioned my name. Based on that, then Richard later asked me to join the LMB on a tenure track group leader position. So I arrived in 2010. So and then the first thing what I did was not use much of the opportunities provided to me by the LMB of hiring other people. I first sat on my own in a room and for 2 years devised the theory and implemented the theory in the computer program, which Carmen already mentioned, which was RELAUN. So the theory behind RELAUN stands for regularized likelihood optimization. And this is because the work I've done so far in Madrid was all based on maximizing the likelihood here. And this is Bayes' law, and the likelihood is the probability of observing data given a model. Now, what we're really interested in is not the likelihood. What we're really interested in is the posterior, which is the probability that the model that we currently have is the correct one, given the data that we've observed. Now, according to Bayes' law, the posterior is actually the likelihood multiplied by a prior term. What is the probability of my model at that point? And you can see that X, the experimental data, is not part of this. So this is a question: what prior knowledge do I have about reconstructions in CRI-AM if I don't have any data available? And then Bayes says you have to normalize this by the probability of observing the data, which nowadays seems to be more dependent on how much money you have rather than anything else. But once the data is observed, this becomes a constant which can be taken out. So all that we'll be doing is I'm going to supplement the likelihood function that I was optimizing at the CNB with a prior function. What do I know about my reconstructions? And without any data. And that then gives regularized likelihood optimization, which is what RELAUN stands for. Now, the likelihood function was very close to something that we already done with Jose Maria in Madrid in Fourier space, and I'll show some equations, but I won't go into the details. The million-dollar question then becomes, what do I know about CRI-AM reconstructions if I haven't got any data yet? And that's the prior, and the prior that we settled on is something that was tractable computationally, but perhaps seemingly not that information rich. We just assumed that the signal is Gaussian distributed and independent as well in the Fourier domain. And then we limit the power of higher spatial frequency in the Fourier domain, basically imposing smoothness on the real space reconstruction, thereby discarding or down weighting a lot of possible solutions that would be very noisy in real space. Now, that all combined, if you then go through your math, you get and you optimize this target function using an algorithm called expectation maximization. You boil down to an algorithm that optimally filters your reconstructions and does the alignment optimally filtered as well. All these filters are calculated automatically from the data themselves. So this reduced a lot of onus that previously relied on the experimental cryo-electron microscopist to tune weights with low-pass filters and Wiener filter parameters to keep overfitting at bay. Now all this was automated and part of one big Bayesian framework. All that was implemented in RELAUN, and as Carmen says, just as Jose Maria is a great proponent of this, and probably this is where I picked it up from him, we keep being a very big proponent of open science, and we distribute all our code under open source licenses. And if you do follow me on Twitter, then you'll notice that sometimes I rant about this, and that's because I feel very strongly about how open software accelerates science, particularly nowadays in a field of CRI-AM where commercialization, closed source, patent protection, etc., are on the rise. Now back to Richard. Richard in 1995 promised us that based on the fundamental calculations of the main limiting factor of electron microscopy, which is radiation damage. The biological samples get damaged by the electrons that we use to image. Therefore, we need to limit the number of electrons we use for images, which leads to these very noisy images, the very grayscale image I showed on the first slide. Yet, if you look at the physics limits of that, Richard predicted that you could do atomic resolution, or what he meant by that, resolution, reconstructions to resolution sufficient for de novo atomic modeling of molecules that are 100,000 Dalton in molecular weight, needing only about 10,000 particles. Now, for many years, many people thought Richard was rather the optimist, and perhaps the black cloud in this plot explains why that is. The black cloud here are all EMDB, so electron microscopy database entries. These are structures done by electron microscopists in the 21st century. So they're all from the year 2000 to the year 2010, I think. And the resolution here is plotted against size. So the bigger things are, you can get to higher resolution. But very few electron microscopy reconstructions would go beyond visualizing alpha helices as kind of tubular density. Most structures were characterized by having one blob for each protein domain. Now, in 2013, that changed rather drastically. And here's some of the structures that we and others did in that first year since these new direct electron detectors became available. And suddenly we came to resolutions close to 3 angstroms, where you can see the protein main chain and many of the side chains to good enough detail to do de novo atomic modeling. So what happened? First of all, there was the development of the better detectors. So photographic film, which was used, for example, for the data set by Joachim Frank, basically fails to detect correctly 2 out of every 3 electrons that is put on the sample. So on one hand, we try to carefully limit how many electrons we've put in the sample to limit electron induced damage to the sample. But then on the other hand, we throw away 2/3 of the signal by having a suboptimal detector. And it was the development of direct electron detectors, a new type of chips specifically designed for this purpose, that actually raised that to about 1 out of 2 electrons being correctly detected, or at low resolution for the K2 detector, for example, even 8 out of 10 electrons. Now that the appearance of the new detector then more or less coincided with the appearance of the Bayesian framework software in Reliant. And on top of that, there was now, because these detectors are very fast, you collect movies and you can then correct for movement of the samples that happened when you put the electron beam on them. And all these together then that led to this resolution revolution in cryo-EM, with a sudden jump in the quality of our reconstructions. Now, I think also relevant for many labs was then work that we did in collaboration with Eric Lindahl and his students, Björn Forsberg and Dari Kimanius, who's now a postdoc in my lab. They took our algorithm and accelerated it to be used on these NVIDIA GPUs. And because of their hard work, nowadays you can do these type of reconstructions on a desktop-based computer in a matter of hours or days, depending on how big your data set is. And I think this has been another major contributing factor to the kind of democratization of cryo-EM, which is ongoing in the field. So what is possible now? I think one important thing that has grown even further was the work already started with Jose Maria about being able to separate distinct states. Now having less noisy images and better software to separate states, this has really taken off, and you can now image samples which are in mixtures of multiple states, sometimes biochemically deliberately induced to be so, and then just rely on image classification using these programs to separate out distinct structural state. And this is an example from the Rubenstein lab in Toronto, who separated out multiple different rotational states, for example, of this ATP synthase rotor. And of course, because many of the molecules we look at are molecular equivalence of machines where different parts of larger assemblies move relative to each other as an intrinsic part of their functioning, being able to separate those distinct functional state is very important for structural biology and us being able to understand how these machines work. Now, that the methods I spoke to about before, the 3D classification then tries to separate the data set into a discrete number of distinct structural states. But that, of course, is not true for many molecules where a more continuous movement of flexing is the case. And to be able to deal with that, Takanori Nakano and myself implemented this multi-body refinement approach in Milan, where we divide large macromolecular assemblies into multiple independently moving rigid bodies, which are then refined separately from each other for each part of the particle. So rather than assigning one set of orientations to each particle, we now divide it. For example, here is a spliceosome particle divided in 4 independently moving parts. So we assign 4 different relative orientations to each of these parts of each of the spliceosome particles. And that then leads to a major improvement in resolution in the parts that move. So on the top here shown is the reconstructed density for the so-called consensus model, where we just have all the flexing particles combined into one reconstruction. And especially here at the top, you can see that density is extremely fuzzy. Would be very hard to propose any atomic models of that. But if you now separate it into these independently moving parts, you can then get much better reconstruction, even for parts here at the top, this SF3B domain, which moves a lot, which becomes resolved well enough to fit in atomic models. And not only can we make better atomic models for parts that move, you can then also analyze all these relative orientations and make movies from them to then see what kind of motions are actually present in this type of machine, and then those could be informative on how they work again. And this is one of the movies, for example, that we calculated for the spliceosome, where the top part here, this SF3B and this helicase domain, seem to be moving together, and there's other components in the data where they move separately. And it's then up to the experimentalist to interpret these type of movies and see how they correlate with the functioning of this machine. Now, in 2020, this work was actually finished in the attic of my house while all locked down because of the COVID pandemic. This is work that was done by Takanori Nakano, postdoc in my lab, in close collaboration with Abhay Kotecha, who's a scientist at Thermo Fisher company in Eindhoven, where all the big Titan Kraus microscopes are made, the ones that many of us now use routinely every day. So it was again the combination of development of new cryo-EM hardware with developments in cryo-EM software that allowed us to make a major step forward. And on the hardware side, there was the development of a cold field emission gun, a more stable energy filter, and the latest generation of this direct electron detector called the Falcon 4. And on the and just to explain what the effect is of the cold field emission gun, I have to briefly mention to you chromatic aberration. This is something that can happen in optical imaging too, but it happens a lot in electron microscopes because the lenses we use, the objective lens, is not very good at focusing electrons that have different energies at the same point. So how much spread of focusing point you get from different energetic electrons is called chromatic aberration. And this is just an example from an optical image where you can see that some colors in the image are focused distinct from other colors. Now, if you have a chromatically aberrant objective lens, then what you should do is try to limit the spread of energy of the electrons that you use for imaging. And that's where the cold FAC comes in. So if you use a standard XFAC field emission gun to generate electrons, they'll have 0.7 electron volt energy spread, which is not bad over a absolute energy of 300,000 electron volts, but still the cold field emission gun that reduces that by more than half to 0.3 electron volt. And if you then calculate what the effect of a given chromatic aberration in the objective lens is, then you can see if you would use an XFAG gun, then at 1 angstrom resolution, most of the signal is actually gone, whereas with the cold field emission gun, you have more than half of the signal left. Now, the energy filter it's down here separates out some of the inelastic scattered electrons which happen here in the sample, which contribute noise to the images. They lose energy, and these are then separated out by the filter, and this was a much more stable filter. And then this was combined with the Falcon 4 camera, which even further increased the signal to noise ratio, now about detecting 2 out of every 3 electrons over a wide resolution range. Now, this was then combined with software developments by, in particular, Jasenko Zivanov in my lab, who developed these computational methods to correct for optical aberrations that would result from having your microscope not perfectly aligned, and perfectly aligning your microscope when we go to very high resolution becomes very difficult. And it's just being able to then post hoc, based on the data you've already collected, being able to correct these aberrations and then pushes resolution forward a lot further. And these things combined then led to the calculation of this truly atomic resolution apoferritin map. This is calculated at 1.2 angstroms resolution. And because 1.2 angstroms is more or less the shortest distance between any of the heavier atoms in a protein structure, that is then true atomic resolution. And each of the atoms in the protein is now separated as an individual little blob of density, as shown in light blue here. Now, if you then calculate difference maps, and this is work together with Gary Murshidov at the LMB here, where the atomic model is made up only of the heavier atoms, so carbon, oxygen, nitrogen, sulfur, but not the hydrogens. If we calculate a difference map, then in the difference map, we get very nice difference peaks for many of the hydrogens, not all of them, some of them are missing, but you can now really start to investigate what are hydrogen bonding patterns in your protein. And my favorite part of the map is this molecule here, which is H2O. So we have a beautiful blob of density for the oxygen and 2 beautiful difference density peaks for 2 hydrogen atoms. So that's your ordered Walter inside apoferritin. Now, we also, together with Radu Arăcescu, we applied this to GABA-A homopentamers to have a more realistic structure. And using this microscope, we were able to calculate a 1.7 angstrom map, showing that even for very medically relevant molecules, you can also get to very high resolution. And even at this resolution, you can see hydrogen atoms in difference maps. Now, as Carmen said, we've also worked then since 2016 with Michelle Goddard in the neurobiology division, working on amyloid filaments that we extract from the brains of people who've died with a range of different neurodegenerative diseases, Alzheimer's disease being the most common one. Given the time allocation, I don't think I can go into much detail, but overall, I am happy to report that we're having a lot of fun scientifically here at the LMB. So my brief talk here also contains some instructions to speak about my experiences at the CNB. So I have 2 slides on that. And the first one, the point I wanted to make is I'm very grateful to Jose Maria Carazo, who from the very beginning, when I wrote to him from Utrecht saying, you know, I would like to work on electron microscopy, pointed me toward the biggest problem in the field, which was how can we separate distinct structural states from these data sets. Now, almost 20 years later, I would say this still continues to be the biggest problem in the field. A lot of progress has been made, but you know, you only have one life, so you better dedicate it to important problems. And it was Jose Maria who pointed me this way. So I learned a lot about computation. Jose Maria's group had a lot of computer scientists and mathematicians and people trained from all kind of walks of life that I was not accustomed to coming from a chemistry department at Utrecht. So it was a very fruitful environment to learn new skills. And that together with that, Jose Maria also had an experimental side to the group. So there was a good mix of people doing the experiments who could tell you what their real problems were, and the computational people. And this mix, I think, is very important to keep computational developments focused on what really matters to the people who are doing the experiments. And overall, I was very happy at the CNB. It provided a very pleasant and productive environment where, you know, there was good social coherence. Now, on the other hand, I think I do have to mention that because of the personal circumstances that Carmen mentioned at the very beginning in the introduction, I did try 4 years to find a more open-ended type of position, but this also included Ramon y Cajal type of positions in Spain, and was unsuccessful to do so. Yet the LMB invited me to apply to a tenure track position, and then offered me that position. So I think in terms of scientific quality, I think there is evidence to show that the quality was high enough to do good science, good enough to be able to in principle achieve such a position. So that then has to bring me to the conclusion that recruitment in Spain, and perhaps the CSIC in particular, is not driven mainly by scientific quality. And that is partially fault of a wider system of government and bureaucracy that is part of, but only partially so. We're all part of the system we live in. And I think one problem of this system is an absence of feedback loops on quality once people are hired. So that brings me to my conclusion slide. I've shown you how detectors and software developments have led to this cryo-AM revolution. I think part of this revolution was kind of seeded at the CNB, even though this was many years before. In Relaun, both marginalization, as I said, developed at the CNB, and explicit regularization, putting everything in this Bayesian framework, which was new in Relaun, are very important. And one of the key things that Relaun has been very good at is to separate these distinct structural states, both discrete and continuous, although for continuous flex in motions, developments are still ongoing. And then recently we've been able to reach this milestone of true atomic resolution, again through a combination of hardware developments and software developments. And then as a final word about science in Spain, I think it will continue to suffer without more money. I think science in Spain, I mean, this has been a problem for many years. If you compare to other countries, the amount of money that is spent on science is a problem, but it's not the only problem. I think even if there would be more money, you would still have to have a rethink about the role of scientific quality in the whole organization of science in order to make it a real success. So that brings me to my acknowledgement slides. On the left are all people involved in the work I've shown you. I've mentioned many names. Rafael was very important in the development of early Relaun interfaces and how you interact with the user as well. I didn't speak about that. And I'm grateful for all my colleagues here at the LMB now, but also those that I had the pleasure of working with at the CNB about 12 years ago now. Okay, so that's it. Thank you very much. I'd be happy to take some questions.
Thank you, Charles, for this great talk, both on the science and on the reflections about the science system. So I know there is a delay with YouTube, so maybe while we wait for the questions to appear, I'll ask one question myself. So what would you say would be the next step in the democratization of cryo-electron microscopy? I've heard Richard Henderson talking about 100 kilovolt electron microscopes, which are cheaper, but on the other hand, we need the cold field emission gun to achieve the highest resolution. So
yeah, so I think cheaper microscopes for democratization is still the main big step to take, because software-wise, you know, image processing-wise, things have become a lot cheaper, but doing these experiments remains extremely expensive because the 300 kilovolt microscopes are 5 million a pop. So that becomes a cost also with maintenance contracts, etc., that are very hard to bear by many, many institutes. So having a lower voltage microscope, and Richard and Chris have shown that using 100 kilo electron volt microscopes, you would get higher signal to noise ratios based on the physical limitations of radiation damage than at 300 kV. But all kind of things like optical aberrations are more important. So there are limitations, but they've already shown that if you use such a microscope, which in principle would not need to cost more than 500,000 euros, so about a tenth of a cryo's, you would be able to get to 3 angstrom resolution reconstructions routinely, which may mean that a lot of questions that people have all over the world would already be answered at that resolution, because not everything requires atomic resolution. And those samples that would then be perfectly pre-screened. You'd have good grids. You could go to national facilities or international ones, where you just then give that final push in resolution on a much better optimized sample. So I think that would be a good way towards democratization.
Okay, thank you very much. I can see that in YouTube we have lots of thank you messages for sharing your research, for your thoughts and analysis on the problems that we still have in the Spanish science system. What else? Jose Casasnovas is asking, could you please comment on the molecule size and resolution in cryo-EM?
Right, so there is, the smaller things come, the smaller the molecules are, the lower the signal to noise ratio in the images, because there's less scattering potential available to form the signal. So there is no upper limits is not so important. I think it's the smaller molecules that matter most. And I think we're currently, sub 100 kilodalton is well possible. The more you go below 100 kilodaltons, the more difficult it still is. And I think the more help you'll need from experts if you're new to this field. So 60 kilodaltons has been done, but I would say that's still quite hard going, and it would need very careful image acquisition and sample preparation and image processing.
Okay, thank you. I cannot see any more questions coming, so
that's
Mario,
fine. Thank you, Carmen. Thank you, George. It was a great pleasure to hear you. It's really fantastic talk. Thanks a lot.
Thank you.
Then we move now to the Department of Plant Molecular Genetics. Carmen, it's your turn.
I think Susanna has to open the video. I cannot open the video. Susanna. No, wait. No. Okay.
Now you are on the video, Carmen, sorry.
Now it's okay. Can you hear me? Yes. Can you hear me?
We can hear you and we can see you.
Yes, yes, very well.
No, iniciar my video. Okay, I don't know if you see me, but anyhow, it is a pleasure for me to introduce our next speaker, Salome Prat. And let me start by telling you that Salome is an outstanding scientist that has made major contributions to the field of plant development and signal transduction. Salome has been working here at the CNB for 18 years. And for us, it's not only a great scientist, it's not only a great colleague, but it's also a very good friend that we always welcome here in Madrid, and we always welcome here at the CNB. She's now working in Barcelona at the Center of Research for Agricultural Genomics, where she moved 1.5 years ago. And she's establishing again her laboratory in that center. Salome, along her career, has achieved a high international visibility. She has published her results in more than 100 publications, many of them in high impact journals. She has been invited to the most important international conference, and she has participated in many international consortiums. One of the main merits of Salome is that she has carried out her research in a very difficult experimental system such as potato, where she has contributed to understand the mechanisms controlling tuberization and the effect of environment conditions such as light and temperature in this process. She has also worked with an easier model system such as arabidosis, where she has contributed with many interesting results in the study of hormonal control of plant development, and she has identified and contributed to the identification of key regulatory proteins. As I said before, Salome has been working at the CNB for 18 years, but let me add that she is a very dedicated scientist. And if we could count the number of hours that Salome has been working here at the CNB, I'm sure that she will be the winner. And so she is probably the person that has worked more hours here at the CNB. So in the name of the CNB, Salome, we want to thank you for your contribution during all these years and all these hours. You are always very welcome here. And now you can have, I think you have 30 minutes for your presentation.
Okay, thank you, Carmen, for the nice introduction. And it's true that I spent many hours in the CNB, even just before leaving, no? Just a few days before my moving, I got with the Filomena, I was just packing, so I spent 3 days here in the CNB without being able to go home. Okay, so I'm going to share the screen. Okay, I hope you see it. Oh, you see my screen?
Yes. Yes.
Okay, so what I would like to talk today is about the thermosensory role of COP1, that is an E3 ligase that was always associated with light signaling in plants, as revealed by a mathematical model. And I choose that talk because this mathematical model was done in collaboration with the Systems Biology Department of the CNB, and this is a work that we would not have been able to do by ourselves. Okay, this is the Crack Institute where we moved since January last year. I moved there with only one student from the CNB, Luis Miguel Luengo, that was finishing the PhD. And now, well, we had to start our group, but okay. But now, after 1 year, more or less, we managed to resize our group. Okay, so in principle, we always associate complex biological models with movement. Okay, we think that animals that are able to move are more complex than plants because plants spend their life fixing soil. But I have to say that it's just the opposite. If plants live fixing soil, they need to adapt to the progression of the different seasons, and they need even to advance the arrival, for example, of winter. How do they do that? They do that because they sense the light. They have a completely different development in the presence of that, or in the absence of light, or in the presence of light. And this is exemplified here in this slide, in which you see how a young seedling develops just after germination if the seeds are germinated in darkness or in the light. Light is also integrated with an endogenous system that is the circadian clock. And the circadian clock has a 24-hour rhythm, is a loop of repressors, and altogether allow the plants to sense the day. Okay, and typical differences between what we call a diurnal response or a circadian response is that the diurnal response, they are set in phase by light. They need light. If we change from diurnal conditions to continuous light, the cycling of this response is lost. Whereas with the circadian regulated responses, they keep cycling even with transfer or with switch from diurnal to continuous light condition. And for example, the genes that show this behavior means that they are direct outputs of the clock. And I will talk a lot today about the direct output of the clock of one of the repressive loops of the clock, that is the evening complex, that it exerts its action at the beginning of the night. Okay, so as I was saying, light is fundamental for the plants, not only for photosynthesis, but also for their development. Actually, the plants have different photosensors in the blue wavelength and in the red wavelength. And in the red wavelength, the most important ones are called the phytochromes. And they have a particularity that they are able to change between an active and an inactive state. So when they absorb red light, they switch to an active state that is called the PFR form, and this form can revert to the inactive one upon far red absorption, and also spontaneously in darkness. These activated phytochromes are controlling all the or many different responses during the life of the plant, and the one I'm going to focus today a lot is what we call photomorphogenesis. Okay, that is the adaptation of the plant to the light conditions. As I was showing before, the response is very different when the plant germinated in darkness. The hypocotyl or embryonic stem, it grows very fast, and the reason for that is that the plant needs to, or the leaves needs to reach the surface of the soil to be able to start the photosynthetic life. Okay, when the plant reaches the soil and sees the light, the chloroplast gets differentiated, and this plant is able to start photosynthesizing and be autonomous. It does not depend any longer of the reserves in the seed. But if it's not able to do that on time, then the seed reserves will get exhausted and the plant won't be able to survive. Then, how phytochrome exerts its function and how phytochrome is so important for this very rapid growth of the embryonic stem that is mainly due to cell elongation. It's not due to cell division. Work by several labs, among them ours, had shown that really phytochrome, when it's activated by red light, it migrates into the nucleus, and in the nucleus it interacts with a family of transcription factors that are of the BHLH family, that they are called the phytochrome interacting factors. And these factors are essential for cell elongation. And normally these factors accumulate in darkness, and that's what makes that the cells are very elongated in the absence of light. But in the presence of light, phytochrome goes to the nucleus and induces the degradation of these phytochrome interacting factors, and that's why there is this change in the hypocotyl length between very long in darkness and short in the light. Okay, and when you don't have this cell elongation or elongation promoting factors, what you see is that the seedlings are short also in darkness, as if they would have received light. But phytochrome makes also a second function in the nucleus, that is inactivate an E3 ligase that is called COP1. This E3 ligase is very important for photomorphogenesis and actually, or for the transition between etiolation and photomorphogenesis and a green plant. And what it does in the nucleus, this E3 ligase, is to degrade a lot of transcription factors that are promoting the response to light, and that they negatively regulate the PIFs. It has been that many of the targets of this E3 ligase, either they make complexes with the PIF transcription factors and inactivate them, or they bind into the promoter of the genes that are regulated by the PIFs into the same recognition elements and do not allow the PIFs to induce this elongation response. Actually, the work by many different labs found that really there is a central module for plant growth that is regulated by the PIF transcription factors and by a second family of transcription factors that are called the BESBISR1 that were identified as factors that were signaling the hormones brassinosteroids. And what we know is that this complex is central for activation many genes, or a lot of genes that are involved in auxin signaling and in cell wall loosening. And you need to loosen or to make the softer the cell wall of the plants, otherwise the cells, the vegetal cells are not able to elongate because the cell wall is hindering elongation. And these factors are integrating the signals for many different hormonal pathways and for life. Okay, a very important response that has been found also is that when young seedlings are grown under elevated ambient temperature, so that we are talking 6 degrees over the normal temperature that for Arabidopsis is considered 22 degrees, what one can observe also very drastic elongation of the hypocotyl. You see that these plants are behaving as they would have less light. And it was found, this response is very important to cool the leaves and to protect the meristem from the warm soil. Otherwise, this meristem would get damaged and this plant would not be able to survive. It was found that this elongation response is again mediated by the PIF transcription factors. In this case, it was identified 2 members of the family, the PIF4 and PIF7. And also work by 2 independent labs found that the sensor of temperature, or one of the main sensors of temperature in plants, is again the phytochrome B, okay, the photosensor phytochrome B. This was independently found in 2 laboratories, in the laboratory of Jorge Casal, because he was analyzing the photobodies. So in phytochrome B, in the nucleus, form complexes that are called photobodies that are thought to be the active sites of phytochrome B, and is in these sites where it interacts with the PIFs and the COP1. So they found that the formation of these photobodies is in part disgregated at elevated temperatures. There are much less photobodies, and also more phytochrome B is diffused in the nucleus, and actually measuring the dynamics of these changes, they were able to calculate, they were able to demonstrate that elevated temperatures, what they do is to speed the spontaneous reversion of phytochrome B in darkness, and they were able to calculate the constants of this reversion. And also there was another different lab, one of Philby, was studying what are the genes that are induced at elevated temperatures in Arabidopsis, and they found that these genes share many common targets with the phytochrome, with the ones that are misregulated in phytochrome mutants. So in this way, both laboratories were able to demonstrate that really temperature is sensed in the plant with the same receptor that senses temperature. So that means that these 2 external signals or environmental signals undergo a crosstalk already at the sensor level. Okay. Also, it has been reported that COP1 accumulates in the nucleus at elevated temperatures, and probably that's due to the thermosensor function of phytochrome, because phytochrome is less active and then is not able to inactivate COP1, that is one of the targets of phytochrome in the nucleus, and then we find that COP1 accumulates. Another important sensor for temperature in plants is the evening complex. This one of the circadian clock loops that we had demonstrated that is regulating the diurnal, is responsible of the diurnal expression of the phytochrome interacting factors. And in our lab, we also were able to show that one of the components of the evening complex, that is the ELF3 protein, is also able to regulate the P4 transcription factor at the protein level by protein-protein interaction, and this interaction plays a very important role in the response to ambient temperature. And the response to ambient temperature is very important if we want to understand or to select plants that would be more tolerant to climate change. But we have learned that this response is very complex because they play a role at least for major regulators, that is phytochrome B, that, as I just mentioned, is a photosensor and a thermosensor. It plays a very important role, the P4 transcription factor, that is the final effector of the cell elongation in the plant, and it plays also a central role, the COP1 E3 ligase, that is one of the targets of 5B, but itself it regulates 5B, and it has been shown that this E3 ligase is important for an increased activity of P4, not only because it degrades many of the antagonists of these transcription factors, but also allow or mediate the degradation allow an accumulation of P4 in the nucleus at elevated temperatures. And probably it does that through the regulation of L3, that is one of the components of the evening complex, that is a direct repressor of P4 transcription by the clock. So this response was very complex, and just by analyzing the function of these different components was really very difficult to understand how this wheel, in which we found that these four major components closely regulate each other, was very, very difficult to predict and understand how the response to temperature is regulated. So then for that, what we did to understand this response, we started to work in collaboration with Saúl Lares and Pablo Catalán in the Systems Biology department of the CNB, whether they could help us to model this response. What we could provide for the modeling was that we had measured the hypocotyl lengths of several single mutants and double mutants for these four main regulators, and we knew a lot of what was described about how these regulators are controlled by environmental conditions. So then Saúl and Pablo developed a mathematical model that is based on different equations that they are calculating the activity of phytochrome B in the cell by considering its activation by light, the dark reversion reaction that we know is stronger at elevated temperatures, and also the described role of COP1 in mediating degradation of this photosensor under extended light. They calculated also the accumulation of the evening complex to L3 that we know is regulated. It is a circadian regulated, but it's also we had demonstrated that COP1 and 5B play an important role in regulating the stability of this protein. They arise another equation that was calculating how the accumulate in the cell, how they are transcriptionally regulated by the complex, and how the protein is either stabilized or destabilized by phytochrome B, a function that was predicting the biological activity of COP1 was described that COP1 is translocated into the nucleus at elevated temperature. It plays a role in degrading itself, and it also plays a role in inactivating 5B, and a final equation in which they were calculating the growth based on the biological activity of PIFs that we know is regulated by L3, 5B, and COP1. Okay, what we could provide into the model is that, as I was saying, we were measuring the apocotyl length of the different mutants, phytochrome B, COP1, the double mutants, the L3, and we were measuring the apocotyl length of these seedlings under different day lengths, so under continuous darkness, continuous light, and different conditions. Data we used fitting the model, that the mathematical model that it was generated by Saul and Pablo. And here I'm showing the results of all the apocotyl length, apocotyl growth data we had gathered to fit the model, that these are the points, and the continuous line are the predictions. Fitting the model was making for the growth of these different genotypes. And as you can see, the model was able really to capture the growth of these many different backgrounds. And not only that, but was able also to predict the growth of new mutants that we had not yet generated, and that we didn't know what was the growth phenotype of these different mutants. One important outcome of this mathematical model was that, whereas it was accepted by the community that COP1 has a function in sensing temperatures that is completely independent of light, it was demonstrated that at elevated temperature, COP1 accumulates into the nucleus, but this response seemed to be completely independent of light or diurnal timing. The mathematical model was predicting that the activity of COP1 is saturating in darkness, so COP1 accumulates in the nucleus in darkness, but is really limiting or is much reduced in the presence of light, and is under these conditions in which it is reduced when it's especially sensitive to temperature. Actually, the mathematical model, if we predicted the function of these E3 ligases, this E3 ligase in darkness and continuous white light, and what would be the phenotype of Arabidopsis plants that would be either overexpressing this E3 ligase or a knockout mutant for this E3 ligase. The model was predicting that it doesn't matter if we overexpress the E3 ligase. In darkness, the plants will grow the same, whether at 22 or 28 degrees. Okay? So that the function of this ligase is saturating and is not sensitive to temperature. But under continuous white light, it was predicting that an overexpression of this E3 ligase would lead to a much stronger thermal response. To the plants would be much more sensitive to temperature. We decided to test, whereas when we would have, if we don't have this E3 ligase, the plants are dwarf and they do not respond at all to temperature, not in the light, not in darkness. Okay, this is also an important outcome of the model, because it says that this E3 ligase plays an essential role downstream of phytochrome B and downstream of other thermosensors as the clock or the evening complex. Okay, when we experimentally validated the prediction of the model, we found that actually the COP1 overexpression lines, it doesn't matter the levels of overexpression they have. We tested different lines with different levels of expression of the COP1 ligase. In darkness, the plants are very elongated and they hardly respond to temperature. But when we draw these lines under continuous light, we see that the response to temperature is linear with the levels of overexpression of the E3 ligase. And also, we were able to demonstrate that this saturating response or activity of the COP1 E3 ligase in darkness is not due to an accumulation of the protein, because we see that in darkness the levels of this protein are even slightly reduced compared to the levels in continuous white light. And probably the reason for that is that the E3 ligase is able to auto-ubiquitinate and degrade itself. And probably this higher activity in darkness makes that at the end we have less of this E3 ligase. Okay. Another important prediction of the model was that if we would generate double phytochrome B COP1 overexpressor lines, so the plant would have a constitutive response, a constitutive elongation response, as if they were in darkness. So they would not respond to day length, so to light, and they would not respond to temperature. When we experimentally tested this prediction, we found a response that was quite similar to what the model predicted, was not perfect. Okay? You see that these plants still respond to light and still respond to temperature much less than wild-type plants, in which the curve would be like this. They are partially insensitive, but they nicely resemble to what the model predicts. We tested why the experimental data is similar to the model, and with a reason that probably one of the major reasons for that is that in our mathematical model, we didn't include the function of other photosensors like the cryptochromes, that are the photosensors that sense blue light. And studies in other laboratories had shown that cryptochromes play also a very important role in regulating the activity of COP1, because cryptochromes are able to bind into the target recognition site of this E3 ligase and block the activity. We tested that just by comparing the growth of this double mutant and the continuous red light and continuous blue light, and we see that under continuous red light, in which only phytochrome B would play a major role in the response to light, the plant resemble a lot to what the model was predicting. Okay, they show a constitutive response, whereas in continuous white light, the divergences with respect to the mathematical model are exacerbated. They are increased. So that means that if we would need to upgrade our model to include the cryptochromes, and probably then we will get a very, very nice prediction on what would be the response to temperature of the different plants, of different crops. What I want to show here is a prediction made by the model, in which it was used to predict what would be the thermoresponse. So that means the difference in growth between plants' growth at 22 degrees and 28 degrees of plants that would be grown under different wavelengths, and the role that they place, the major regulators I showed to you into the response, the phytochrome B, the COP1 or the L3. And as you can see here, the phytochrome B is a negative regulator of thermomorphogenesis, because we see that to have a strong thermomorphogenic response under continuous white light, what we have is to have a reduced level of this regulator. Okay, whereas we have the opposite in COP1. COP1 has a positive role in the thermomorphogenic response, and we have a greater response the greater are the levels of COP1. Also, the model predicts that COP1 is essential to the response to temperature because in a COP1 mutant background, so if COP1 is inactive, it doesn't matter whether we have more or less 5e or we have more or less evening complex, these plants are not able to respond to temperature. And also it allows us to make very nice predictions respect the interaction that there is role 5e plays a role in modulating the activity of COP1, and also it predicts that L3 is also the evening complex, or at least the L3 protein is modulating the activity of the COP1 E3 ligase. These findings are new, are predicted by the model, and what we are involved now in the lab to really demonstrate that these predictions by the model are really true. But what I want to highlight here is that this mathematical model is extremely useful for breeding because it tells us which would be the best allelic combinations to get thermotolerant plants in crops require short day conditions or crops that are adapted to dayland conditions to be able to be resilient to climate change. Because what we see is that thermomorphogenic response is strongly dependent on dayland. Okay, so what are the remaining questions? Okay, one, and what we would like to tackle, or we started studying since we are in the crack and probably will be the project of the future of in our lab in the next years, will be to understand what is the mechanism by which temperature promotes the accumulation of COP1 in the nucleus, because it is shown that COP1 normally accumulates in the nucleus in darkness, but in the presence of light excludes the protein from the nucleus, and that's what it allows to initiate this photomorphogenic response. Okay, so we would like to know this mechanism and really to identify how it is regulated and how temperature impacts in this mechanism of nucleus cytoplasmic partitioning. We are also studying what is the exact role of COP1, why COP1 is required for an accumulation of the cytochrome interacting factors in the nucleus and for plant growth, and the prediction of the model that they were saying that L3 has a main role in regulating the activity of COP1. Okay, for the nucleopartitioning of COP1, we are making use of a particular mutation of the COP1, that is the COP16 mutation that was identified as a thermotolerant mutation, and because this plant has a 22 degrees of phenotype as the COP14 mutant and in the light. What we see if we grow it in darkness, and at 28 degrees shows a response that is the same as of wild type plant. And actually, this mutation is a mutation in a splicing site that it makes an insertion in the nuclear localization signal. So what we think is that we can make use of this mutation to understand how temperature is regulating the nuclear partitioning of this central E3 ligase in light signaling and in growth. And what we were able to do is to connect this nuclear partitioning with brasinosteroid signaling, because we know if we inhibit brasinosteroid signaling in the COP1 E6 mutant, what we find is we suppress this thermosensitive phenotype. So this response is telling us that really brasinosteroid signaling plays a very important role in nuclear partitioning of COP1, and we could really demonstrate that, because if we analyze nuclear levels of high-5, that is one of these photomorphogenic promoting transcription factors that is a target of COP1, we find that this transcription factor gets more degraded if we treat the plant with brasinolite, and it accumulates in the nucleus if we inhibit the brasinosteroid biosynthetic pathway with the inhibitor brasinazole. Okay, so this
sorry, Salome, is 1 minute to finish. Okay.
Okay, I'm almost finished. So this is telling that really this plays a very important role in this regulation, and actually it is data in the literature that is saying, was reporting that the COP1 is able to directly interact with WIN2, that is a master regulator of the brasinosteroid signaling pathway, and we had shown in the lab that really the PIF transcription factors are involved in the modulation of the brasinosteroid biosynthetic pathway. What I want to show you here is that we have our hypothesis is that the BIN2 kinase is able to directly phosphorylate the COP1 and modulate its capacity to degrade its target. And we have seen that really one of the phosphorylation sites is in the target recognition motif of the COP1 that is known to recognize a BP dimeric sequence in its targets, and actually what we see is that one of the phosphorylation sites is just in the target recognition sequence. So we think that really phosphorylation will change the recognition of the target and will change its response to temperature. And this is what we are trying to do now. Okay, here just I would like to highlight temperature with climate change is increasing. We have superated the threshold that we had said not to overcome the 5 degrees by 2025. We have far over this threshold. So really we will need to understand how a plant responds to temperature and how temperature affects the adaptation to the seasonal adaptation if we want to keep growing crops in different environments and not to have shortage of food for a growing population. And here I would really would like to thank my team in the CNB for many years, and especially the work that they showed today on the model for fitting the modeling and the COP1 brasinosteroid sensitivity that was done by Cristina Nieto and Luis Miguel. And really thank you for all the great time I had in the CNB, and really I want to convey that in the genetic plant molecular genetics department we had always a very nice atmosphere. We are friends, the different groups help each other. I thought that this was the normal thing, but I want to say that really no, this was something very special from the CNB department. And thanks.
Okay, thank you, Salome. I don't know if you can hear me, yes?
Yes.
So I have to wait now to receive the questions. I don't know, I think Susanna has to send me the questions if there is any question, but in the meantime, Salome, you have shown an example of collaboration between the system biology department and your laboratory. And I wonder, you can tell us really how difficult it was to really understand each other. I mean, how difficult it was for you to explain them what you want from them, and how difficult it was for them to understand, and vice versa. How difficult was, this is one question, because really, I mean, it's easy to tell, but I can understand that it's very difficult really to put together. And the second thing is that if you are planning to keep working to improve your model and to integrate other components.
Well, certainly it was difficult at the beginning. Okay, we had the advantage that Pablo is biologist. Is not just a mathematician. Okay, it's true that he got specialized in mathematical models, but as a formation, he's biologist, and this was very easy because he was really able to understand. Because imagine, I mean, we were going with a very complex model in which people has been normally trying to understand the function of these regulators just by measuring the hypocotyl. Okay, actually, in plant biology, there is like there is two difficult tasks or difficult projects to work with. One is the clock, because that means that really you need to spend time courses of 24 hours so you don't sleep at all. And the other one is light signaling, because people is measuring hypocotyls. So that means that you need to use the image J to measure hundreds of hypocotyls. What is I have to say, is extremely boring. Okay, but and that's the job. You say, well, what do you want to do? You prefer to measure hypocotyls, but on the other hand, is very informative, because in this way we were able to learn how these different regulators work. Okay, but we had to explain them. The PIFs are the main effectors. These are the importance, but they are regulated by everything. Okay, they are regulated by the clock. They are regulated not only ELF3 as a part of the clock by direct interaction. They are regulated by COP1. The first times we were meeting, I was thinking we will never reach to anything. But on the other hand, after fitting the model, it was impressive. I have to say that this model is impressive, because what the model predicts, it fits perfectly with what we see or experimentally. And if it's not perfectly, we can understand why it's not perfectly, because in our model is still missing some information.
But are you planning to continue with the model, or you are
Yeah,
going to
for
give
sure. No, we are planning to continue, because, for example, in the model, L3 that we know is one of the components of the circadian clock. It was modeled just to force that L3 was transcribed in the evening, and also at night and not in the day. So it had at peak in the night, because experimentally that's what we had seen, because the model was able to accept other patterns of expression of L3. Okay, that this was something that, to me, we had big discussions with the modelers, because I've seen that mathematics accepts a lot more combinations than biology, okay? In the sense that many things that mathematically are possible, in biology are not possible at all. And this was a big discussion always we had when we were talking with the mathematicians, that they were saying, no, but a different configuration of the model is possible. And I was saying, no, well, this was making me very nervous, because I was thinking, it has to be just one and only one, and it's not true. Mathematically, there are several combinations, but then we can provide the input that we have from biology. And in the case of the clock, we have very nice data derived from a luminometer assays. So if you fuse to the promoter to the luciferase, you can have a very nice record on the exact moment of the day that the genes are expressed. And we can use this data to exactly fit the model. So, for example, we can fit the equation of L3, we can fit the equation of COP1, and in this way, then the model will be able to really predict, okay, then you can go to the model and say, well, I want a plant that is not able to sense temperature in its natural habitat, that is let's say 16 hours light, okay? Well, in the moment, the cultivation season fits with summer, and this is the hours that it receives. And it will be able to say, okay, you would need to look to work with a mutant in this regulator or an overexpressor or that, and this is going to assist breeding a lot. So we will, yeah.
I think we have to stop. There is not really question. Many people is giving you thank you and telling you hello, and so I think you will have chance to see them all
Later.
of them later during the lunch time. So thank you again, and we enjoy very much your talk.
Okay,
Bye.
thank you for inviting me.
Bye.
Thank you very much, Salome, for this amazing work as usual. That's really fantastic. Then we have time for a break, and we need to come back at 11:50 just to start again with the meeting.
Okay.
Then, now we disconnect. That's fine. See you in 10 minutes. Well, good morning again. We are going to restart with this workshop about the 30th anniversary of the CNB, and now the time is for the Department of Microbial Biotechnology, and it's Silvia Llora who has now to start.
Okay. Hello to everyone. Can you see me, hear me? Yes?
Yes.
Okay, thank you very much. So today is a great pleasure to me to introduce all of you Victor Parro, who was probably one of the first PhD students in the CNB, yes. So he moved already to the CNB in 1992, because at that time he was making his PhD thesis with Rafa Pérez Mellado, who maybe some of you know. He was a research scientist in our microbial biotechnology department for many years, since he died several years ago. So, so Victor is one of the first settlers in the CNB, and I think he has probably many stories that he can tell us about the first days in the CNB, how difficult it was for them to make the laboratory run properly and so on. So at that time, he was already interested in biotechnology and microbiology, and he studied in agarose of commercial use. And this was an agarase, of commercial use, an enzyme that degrades agarose. And this was part of his PhD thesis that he defended in 1994 in the Autónoma University of Madrid. After his PhD thesis, he decided to stay at the CNB, working together with Rafa Pérez Mellado as a postdoc and in many different topics, and then protein secretion, because he always has had an interest in biotechnology. So I think we can say Victor likes challenges and setting up new things. And so that in 2000, he moved to a very recently founded institute, which is the Centro de Astrobiology, that was founded at the end of 1999. So that again, moving in a new institute, he had to set up a new laboratory, makes the things run. So the Centro de Astrobiología is a joint center between the Spanish National Research Council and the National Institute for Aerospace Technology. And at that time, the CAP was the first astrobiology institute outside of the United States to be associated with the NASA Astrobiology Institute. The research in this institute is focusing in understanding how life can develop in galactic environments and developing tools to analyze this. And here Victor has many important contributions analyzing at the genomic and transcriptomic level microbial life in extreme environments that we have in Earth as a model, and how can be life in other planets as in Mars. So that Victor has studied microbial life in many parts of the planet, not only here in Spain in the Rio Tinto liver, but also, for example, in the Atacama Desert in Chile. And I think he will tell us many interesting stories about that. But Victor is a scientist, is a microbiologist, not only interested in understanding life, but also in developing technology. And as such, he has many patents and developed many tools and detectors to study life. And I would like, among them, I would just highlight some of them as the SOLID module, which is a detector for, as the name says, is a detector of signs of life for planetary exploration that could be a candidate to go in the next mission on Mars, or the CMOL detector, an instrument to search for biochemical complexity on Mars or also on icy moons. But not only, he develops the technology not only for exploring outside in the planets, but also, for example, he recently developed a detector, a microarray-based detector for detecting antigens against SARS-CoV-2. So finally, I just would like to mention that in addition to this very successful work as a microbiologist and as a technology developer, Victor is, since 2019, the director of the Astrobiology Center, where I guess he also has many challenges to settle. And among them, for example, the COVID pandemic in 2020. So it is a very great pleasure, Victor, to have you today here to tell the stories that you will have. I think you are going to give us a very interesting talk about many things. So thank you again for accepting our invitation of our department. And now is your time.
Thank you very much, Silvia, for your words. You have made an extensive summary of my. Let me do it. How this is work. Oh no, I think I have to share the screen. Can you see the screen now?
Yes.
Okay. Now?
Perfect.
It's okay. So thank you very much, Silvia, for your words. Yes. It's a pleasure for me to be here at the CNB, where I spent so many years, and to meet some friends and colleagues in this special day of the 30th anniversary of the CNB. First of all, well, Silvia has mentioned it, but I wanted to honor the memory of my mentor and the PhD director, Rafael Pérez-Mellado, to whom I spent a lot of time, and also, as Silvia said, was one of the first settlers of the center of the CNB. In fact, we were at the CNB before the CNB was built and really in operations. But he moved with, we moved together with Paco Malpartida and Victor Rubio. We were also the first, at least in our department, were the first that went to the CNB. So, well, there were tough moments, like Silvia said, but there were some funny moments too, and some happy moments too. This was my birthday. It was because I was in the center, that's all. And we were so young. Jose Luis is over there, and Trini and others. But we always take care of the finest and high quality reagents for the laboratory, as you can see here in these celebrations. But also funny and claiming moments. There was some. We have time for claimings. You know, the always eternal claiming in Spain for getting this, I would say, asymptotic. I don't know if sometime we will get to this 20% of the GDP for science. But okay, we did a funny show in the Galileo Sala. Not everybody can say that has shown his back, let's say his back in nature, but we have seen this funny picture in nature in those days. Later, Silvia said, I had to change, to move, to find new challenges, and in that time inspired and encouraged also by two of the big researchers at the CNB, that is Victor de Lorenzo and Alfonso Valencia. They encouraged me also to move to another adventure. That was the Centro de Astrobiología. The Centro de Astrobiología, as Silvia said, was, I think still we are practically the only physical center devoted to studying the life in the universe as a whole in a transdisciplinary setting and in a physical institute. So, but I wanted to show you a glance of what astrobiology is and what astrobiology means. Well, the hypothesis under the astrobiology is that life is a consequence of the evolution of the energy and matter in the universe, and as a consequence of this, the presence of life in other planetary bodies would be a consequence of that. So finding life is an obsession. Finding life outside the Earth is one of the way to demonstrate this hypothesis. But for that, we need the many disciplines to work together. So it's a multidisciplinary field, but they also need to do transdisciplinary work. That means that they had to join all the effort together, from the cosmology, astrochemistry, planetology, chemistry, biology, etc. All of them to study the key aspects of the origin and evolution of life in the universe from the very first beginning, the formation of the first elements, the first galaxies, the first elements and planets and molecules in the interstellar space, the first planetary environment that at the end allowed to develop the complex chemistry that ended up in the origin and evolution and spread of life, at least on Earth. So somehow we are always asking, well, is the carbon-based chemistry universal? So that's another way to pose this question. But it's not an easy task. In fact, there is a controversy even in the definition of life, you know. But well, we can have a consensus, more or less, that life is a self-sustaining chemical system capable of Darwinian evolution. Okay, wonderful. But we know that we need at least three essential components: the metabolic machinery, we need a membrane compartment, we need some genetic templates and inheritance mechanisms, and also we need the Darwinian evolution, for sure. But yeah, if we wanted to look or to search for life outside, we cannot find all these elements together. It's a complicated task. So, for example, how can we detect signs of life? Okay, we can see life. Well, for example, on the left, you can see animals, you can see bacteria under the microscope, and we know that they are alive because we know them very well. We can analyze a lot of chemical and biochemical components in our laboratory, and we know that they come from life. Or we can go to nature and see these bioconstructions were made by ants, or this here that correspond to a stromatolite that had been built by microbes billions of years ago. Or we can go even deeper into the molecular fossils and discover the report the formation of this type of compounds, or opaque derivative, or this one derived from from cells. Okay, yeah, we know them. We know that they are biomarkers because we know them very well here. But what happens when we go outside and we don't know what we can find, and or even if we find just small pieces of these compounds? It's complicated. So Mars is the favorite candidate for detecting life outside the Earth, and for many reasons that I will explain later. So, and in fact, the search for life now is one of the priorities of the, if not the first priority of the space agencies, because yeah, this is an outstanding question, and this is always something that has been in the human memory since the beginning. So this is what you can see here is just a summary of the different missions sent to Mars in the last 2 decades, orbiters that many of them are still operating, or these rovers that you can see in the bottom part. 3 of them from NASA, you see yellow, that are still operating. The one is the Curiosity rover, where we have an instrument that has been developed here in our institute. This REMS instrument is an environmental station. The InSight lander, which also has some environmental station developed by us, and the the Mars 2020 mission with the Perseverance rovers, where it contains also an environmental station that we have developed here in our institute. And there is also some other rovers, like the Chinese rover that is still operating there on Mars. So all those 4 rovers are still working. Well, this is a lander, they are still working on Mars. And what next? Next is in fact, the Mars 2020 rover is the beginning of the Mars sample return program that NASA and ESA wanted to, are working on it, so that, because the idea is to bring some sample from Mars in at the beginning of the 30s. So the idea is to bring sample to the Earth and use all our potential for the analytical capabilities for the analysis of these samples. So the idea is, as I said, seeking for sign of life and prepare for human exploration in the future. This is where all the targets of all these missions. And just to show you, this is the Curiosity rover this is the REMS instrument that we have there and operating since 2012, every single day, and still working on it. And this is the MEDA instrument that is on Perseverance rovers, where we are still operating. Well, we are operating since February last year, and is now started to give an interesting result that is being published in the next months. We know many things about Mars. We know that 4 billion years ago, Mars was containing enough water to cover the surface of the with liquid water nearly to 140 meter depth. We know that in the northern hemisphere, this depth could get even to 1.6 kilometer deep. So, well, we can say that was a water planet in some sense by that time. Okay, but what happened with Mars? This is one of the results from the MAVEN mission of NASA MAVEN mission, where they really reported how Mars is literally losing the atmosphere, 10 grams per second during 2 billion years. That means that that converted Mars in a really barren planet, where high ionizing radiation, UV radiation, and extremely low temperatures made this, at least the surface, practically inhabitable and probably sterile. We don't know. Okay. In fact, the first planetary mission that was looking for life was the Viking missions. They did some experiment that was what is called the lava release experiments that consisted in adding some soil to a beaker that contains several nutrients that were C14 labeled, and just to measure the evolution of gas, just understanding that this was due as a consequence of microbial metabolism. In fact, the experiment was positive. This curve you see on the right indicated that CO2 was produced and the negative control did not produce. But the point was that another instrument in the mission that was a gas spectrometer didn't detect anything, any remain of organic carbon. So this left this result in or enter into a controversy that still today some people discuss about the result, but the point is that nothing was conclusive. And even later, there was another paper that was came out in Science, where they reported a potential microbial fossil from Mars. They made a lot of studies concerning the magnetite crystal inside that resemble those that contain the terrestrial magnetotoxic bacteria. But even though with a lot of studies, still the controversy is still there, because there are many other chemists that say and demonstrate later that this magnetite crystal can also be produced from abiotic sources. So still, again, the controversy. But it was interesting because this works somehow boosted the interest for searching for life and exploring Mars. This, together with the discovery of extremophile microorganisms here on Earth that we know that grow in every part of the Earth where they can find some bioavailable water. So more things about Mars. We know from Curiosity and now from Perseverance that there was running water and relatively big masses of water running on the surface of the planet. You see here on the left these round stones that indicated that it was due to the action of water currents. We also note that that there might be some brines underneath the Martian surface. This is the result from the radar obtained with the Mars Express probe, where there seems to be some brines down to 1 kilometer depth in the Martian surface. But it's interesting because, as we said before, there was a lot of water on Mars, and there might be some subsurface reservoirs that contain these waters. So, and we know there is organic matter on Mars. This is the one of the results from the Curiosity rover and the instrument, the SAM instrument. There is a gas chromatograph mass spectrometer where that detected the presence of organic compounds, but this kind of compound. We're talking about phenol rings and similar polyaromatic compounds, and also thiophene and others, and some small hydrocarbon chains that have been detected in some samples on Mars. But all this organic material can be explained as from a meteoritic origin. We know that meteorites contain this type of molecule even some amino acids. So the fact that we find this type of compound on Mars not necessarily indicated that it come from a biological origin. So still, the question of origin, biological origin is still there. Even it has been detected some methane peaks in some of these experiments. But again, all this can be explained from geological and chemical and abiotic sources. So meanwhile, because the missions to Mars are very expensive and not always is possible, one way to tackle the problem of understanding or to assess the feasibility of life outside is just to study those places on Earth that somehow resemble or are analogous to those environments that we now know that might have been occurred in Mars. As for example, in the first years of the history of Mars, we know that there might have been some hydrothermal environments. Many of them are enriched in iron and sulfur, subaquatic environments, so surface environments, whatever lakes and rivers that we know that were present 3.85 billion years ago. So the idea is to understand these environments, the analogous environment here on Earth, to understand how microbes thrive in this environment, how they interact with the environment, and what remains they can live in this environment, so that we can understand now, or at least to help in explaining the result of the current mission, or to prepare new missions to Mars. And for example, it is known that there are a huge halite deposit on Mars, and we have a wonderful example here on Earth, like the Atacama Desert, and we know in this case that there is plenty of life inside these halite crusts, where you can see this green stuff that correspond to cyanobacteria that are living inside these crusts, making the metabolisms and the photosynthetic activities. We know that there are places on Earth where the microbiology is based on the iron and sulfur chemistry, and they live in the very low and extreme pHs, and this has a strong correlation with some places of Mars that it has been discovered the presence of hematite and a lot of iron and sulfur compounds. So understanding how life thrives in this condition might help us to assess the possibility of life on Mars and to discover life on Mars in the future. For example, we did some studies in some years ago through metatranscriptomics and some other studies, and we were able to identify that, in fact, in the Rio Tinto area, there was a polyextremophile environment with a lot of extremes: pH, heavy metal concentration, salt concentration, high sulfur, high iron, so oxidative stress. So as you can see here, all the red proteins correspond to those involved in pumping these toxic compounds, just flux pumps, some chaperones to control the folding of the protein. We identify some other genes in signaling and biofilm production, and many others involved in repairing DNA, because they are subjected to a very high oxidative stress, and for sure that the DNA must be repaired continuously. In fact, there was a huge increase in the transcription of transpossessed genes and some genes that involve in DNA. We say alteration to try to sort with this stressing environment. So we investigated also how life are inside the rocks, how life can be in the deep of this environment. In a project we have in collaboration with NASA some time ago, we investigated how microbes can live inside the rocks. Here you can see, for example, small clusters or small colonies that are bound to the mineral crystals, in that case pyrite, where they obtain the energy and somehow protections. We understood from this study that life is very heterogeneously distributed, very patchy in this environment, and you might have some rocks where no life at all, and you might have some small areas where there are enriching colonies like this. So it's highly heterogeneous. We discovered for the first time that viable cyanobacteria are possible there, down to 600-meter depth. This picture you see here is some cyanobacterial cluster that were stained with carfish, and we extracted DNA for this sample. We did metagenomic studies, and we were able to identify the main genes that are involved in the different metabolic pathway that these cyanobacteria can be used in the dark. Even they still retain all the photosynthetic capabilities. They should be able to do fermentative and respiratory pathways that I'm not going to enter into detail, but our hypothesis that they are mainly using the hydrogen that is produced by geological sources in the rocks of the interior of this area, and this hydrogen is used is canalized through many hydrogenesis clusters that we have identified, and then through the electron change chains that at the end, in many cases, might be to external acceptors or nitrogen oxidized compound. So we identify also the nitrogen fixation, Calvin cycle, a lot of enzymes that allow them for a rich metabolism. Metagenomics analysis also allow us to identify that, for sure, I'm not going into detail, but to identify clusters that somehow has some metabolic interaction, that is in somehow we think that can behave as all beyond, so in the sense that may behave as a kind of a very primitive association of organisms. So, well, this syntrophic interaction is interesting. That it might be one of the strategies for surviving in such an extreme and sometimes complicated environment. So another environment that we study, as mentioned before, is the Atacama Desert. Understanding the life inside the salt crusts is highly relevant, because we need to understand how life thrives there, and also how the remains of the microorganisms, how these biomarkers are preserved in these environments. So this is the Salar Grande is a big area with 100 meters of pure halite in many places. But we are studying in this case several the cores sample we obtained from a drilling project in an area where they have 40 meters of halite and more than around 60 meters of detrital sediment. So we study here with using a different approach. It was through lipid detection. We extract all the lipids in this area, and we analyze by GCMS and can make different profile like this. We identify different compounds like pristane, phetane, crocetane, so that we can follow the profile along the different depths. So it's a way to understand what kind of remains are living life there. In that case, we identify some different depths in a sample 90 meters, for example, in the halite part and in the detrital part. This is the hopanes. The hopanes are compound. The hopanes are similar, are equivalent to the cholesterol, but in bacteria. They are form part of the members, cytoplasmic membrane of many bacteria, and it's a very good biomarkers, because it has been discovered in samples here on Earth dating 2.7 billion years ago. So it's a very persistent biomarkers in the geological record. But there is an important problem in the search for life and biomarkers in the planetary exploration, and one is the natural constraints that are produced by the techniques itself. We have to be in mind that we are operating instruments that are millions of kilometers apart. So and with a time delay in communication between 8 to 20 minutes. So that means that it's complicated. So far, the instrument that has been sent to Mars for detecting, and to other planets for detecting organic molecules, have been based in the detection of volatiles that have been produced after heating or pyrolyzing the samples. Okay, now there are some other instruments in the Perseverance and others that are planned for the future mission that contain spectrometers like Raman, like laser desorption, another type of instrument that allow to detect organic compound just by spectrometric measurements. And what would proposed several years ago is just, okay, why not use life to detect life? So to use the bioaffinity system, we know, and you know very well, that are in life, that is those based on, for example, immunosensors or whatever enzyme ligand interaction. So what we propose sometimes is just to develop an antibody macroarrays where we produce antibodies and against potential biomarkers and using for mass exploration. And that's what we did, is that we propose just a multiplex antibody immunoassay where several antibodies are immobilized in a microarray. We have now a collection of more than 400, but for the assay, more than 200 start to be problematic. So we are cautious, and we use 200 per assay. The idea is to make a crude extra from the soil sample, filter the most simple as possible, because the idea is to automatize this process, incubate with the antibody microarray, and then add fluorescent mixture of this antibody to allow them to make the sandwich assay. A laser is excite the fluorochrome, and a CCD camera take a picture. The idea is to make the whole entire process we call from the dirt to data. We can quantify this spot. We can analyze and identify those those signals that have intensity below the background, etc. So this way we can identify those compounds that are exactly the same, as very similar to the one we used to produce the antibodies. Okay, so, and in fact it works.
Sorry, Victor, 1 minute left.
Oh, well, oh, sorry. So to go then faster. So it works. This is a sample from the rocks in the Rio Tinto with very nice positives. You see here, we detected a lot of compounds from these rocks. In fact, we detected the first time the cyanobacteria with this from this rock with the light detector chip. In the Atacama, we have a rich of biomarkers in the salt crust. We have developed the instrument that Silvia talked to you before. The solid instrument is a sample preparation unit and the analysis unit with fluidics. We tested in the Arctic, in the Atacama, in many places. Well, the idea is to set the instrument into a mission. We are working with NASA Ames in the Icebreaker concept mission, where solid is part of the payload and is devoted for detecting molecular biomarkers in the Martian permafrost. And yeah, we have, I don't know if you can see this video, we collaborate with them so that we can increase the maturity of the instrument and the concept for the analysis. And just the idea just to be able to have an opportunity for a future mission. It's something that we are trying, but it's not. We have not been able to get it. But okay, this is one. The campaign we did in Atacama in 2019, where we demonstrated the fully automatic and remote operation of solids. This is the instrument you see here on top of the rover, and this is some of the results we obtained. And as Silvia mentioned before, for sure, this technology allow us to develop a relatively quick serological assay for the COVID, that we have done more than 4,000 analysis, and it works very well. But okay, it's complicated to sell because there are too many serological tests now in the market. And just to finish, to say that okay, there is now there are more than 5,000 exoplanets discovered. Many of them start or have the similar size as the Earth, and there are new missions for the future that I'm pretty sure that they will discover much more planets, many more planets, terrestrial like, and for sure, who knows, life might be there in who knows what form, but it's something that still is waiting for us. Many of our colleagues have discovered some interesting compounds in the interstellar space. And okay, just there are many things I would like to tell you, but I guess I know that the time is limited, and thank you very much. This is a picture from Mars, taken from Curiosity rover, where you see on top of the Earth and the Moon. That I like it very much because make us to think what we are and what we do in this universe. Thank you very much.
Thank you very much, Victor, for the talk. I don't know if we have questions. I'm a little bit out of time. Maybe time for a short one. I don't know if it's coming through the chat.
Should I leave the presentation?
Yeah. Well, if not, I have a very short one about this cyanobacteria that you found in 600 meters. How much is the pressure there?
Yeah, that's a good point. Honestly, we don't know exactly what's the pressure there. It's a rock. It's a continental part of the continental crust. Is honestly, there might be some cracks, and perhaps the pressure itself is not too much. But I cannot tell you. We didn't make this measurement.
Mm-hmm. Okay. Well, then we move to the next one. We should keep to the time slot. Or
Thanks,
so that we say bye.
Thanks, Victor. It was a great pleasure to
thank you for you. I'm sorry for the.
Thanks a lot. I hope have the opportunity to know you in person, maybe here at the CNB in other situations. Thanks a lot.
Sure. Okay. Thank you.
Now it's time for the Department of Immunology and Oncology. Then Ana Cuenda has the possibility to introduce Eva. Hi, Eva.
Hello.
Hi. I'm very happy to introduce you to Eva González Suarez in the 30th CNB anniversary, as she took her first step in science in the Department of Immunology and Oncology. And also, I like to think that her stay at the CNB somehow contributed to her development as the excellent researcher that she is now. Just a bit of background about Eva. She obtained her master's degree in biochemistry in the University of Oviedo. And after that, she moved to do her PhD in molecular biology in the University Autónoma of Madrid. And she came here to the laboratory of Maria Blasco when she was at the CNB. During her PhD, Eva did important contribution clarifying the role of telomerase in cancer and aging. After the PhD, Eva moved to Seattle, to the United States, not to another laboratory, but to a company, to Amgen. She was there as a postdoc for 4 or 5 years. And then after this experience in the industry, she moved back to the academy. I think she took the risky, unusual move. And she came back to Spain as principal investigator to the IDIBEL in Barcelona, in Spain, where she was there until 2019. And that year she moved back to Madrid to the CNIO. Her research focuses on the mechanism leading to cell transformation, the metastatic capacity of epithelial cells, and the identification of novel therapies and resistant mechanisms. Research in her laboratory exploits primary cell culture, organoids, a genetic mouse model, patient-derived xenografts, and clinical samples. She has made important contributions to the field of mammary gland development and cancer and breast cancer, highlighting the key role of rank signaling pathway and its potential as therapeutic target. I have to say that rank pathway directly target tumor cells, but also tumor environment, including immune system cells that have the capacity to attack the tumor and destroy it. She's a member of the European Network of Breast Cancer Development and Cancer Committee. She has been awarded multiple national and international competitive projects, including the prestigious Susan Komen Career Catalyst Grant and the ERC Consolidator Grant. She is a world leader in her field, and she is frequently invited speaker in national and international congresses, as well as she has coordinated a clinical trial with rank L inhibitors in breast cancer patients. She has co-authored and authored many articles in top journals like Nature Genetics, you name it. And I have to say that Eva is a role model for all of us, but particularly to our students, since she's a clear example of success on how you can translate basic research to the clinic. And now is the time for Eva to talk about her work.
Thank you, Anna, for this kind and long introduction. I think it's more or less what I was planning to talk on, to comment. So you did it much faster. So I will walk you through this topic. I will share my screen now and a bit of, let's see if I can put it in full screen. Yeah, no, it's ready now. Okay, so yeah, so for me it's a great pleasure to be invited for this anniversary of the CNB. As Anna said, yes, what I learned at the CNB and my experience there has been instrumental for the rest of my career, and hopefully, you know, for the future of it, no? So I arrived in the CNB kind of in between here, and it was a bit thanks to Carlos López Otín, who was one of my teachers in Oviedo, that he encouraged me to do a summer stay at the lab of Maria at the CNB. When I was still at the university, and then after those months and getting to know the people at the 413 lab, I decided to join Maria's lab for my PhD, and that's where I started working with mouse models of cancer to study telomerase, mainly in cancer, a bit in aging. It was a big step because I studied chemistry, so I was a bit far from the mice, but you know, it was so key that I'm still working with many mouse models, as Anna said. And no, that it was a very exciting time because at the time telomerase was becoming, it was hoped to be a therapeutic target, that it's becoming such today. But you know, it was this point where we thought that without telomerase we will cure cancer, and it was very exciting to be there and to author some of the first papers to show the relevance of telomerase in that delicate balance between cancer and aging that has been followed up by the rest of the members with years in Maria's laboratory, coming to really being able to do a super healthy mouse that can live longer. So those were tough years, and I say some pictures on the CNB cafeteria. That was always a good place to do a quick break from the hard work. These years, as for many of you that may be doing their PhD, are very, very important because you establish very strong boundaries with your colleagues, with the time inside the lab and outside the lab. You work hard, but we also party hard. Not only with the people at the DO, but also with the whole CNB, with these going out activities. This was the canoeing that we used to do every year, and it was super fun. And as I said, you work hard, but sometimes you get some nice surprises going to the lab to pass your cells and during Christmas. And getting this super fun image. So definitely it was a very important period of my life, and one of the one that I had more fun. After I defended my PhD, I got the opportunity to join Amgen, Amgen is a biotech company that at the time had bought Immunex, a small biotech. Well, maybe not that small, but that was established in Seattle. And, I knew I wanted to keep working on cancer. I knew that I wanted to go to the US, so they speak English and I could manage with the language. And I love the mountains and the water. I'm not scared of the rain because I'm from Asturias, so I thought Seattle was a very nice choice. And it was a very different environment from academia. I thought that, it was a unique opportunity to be able to do a postdoc in such environment. And I do not regret it. It was a very, very nice experience. Probably very different than doing a postdoc in academia, but very rewarding. And that's where I got to know rank signaling. So this is at the time, like in the DO, many groups were cloning based on homology, different chemokine receptors. In Immunex, they were cloning TNF family members, and they had discovered rank and the receptor and its ligand, which can be in a transmembrane or in a soluble form. And binding of ligand to the receptor through intermediate proteins such as the TRAPs activate downstream targets, mainly in FKB, MAP kinases, or PA3 kinase. And in what it was Immunex and then Amgen, they had developed denosumab, an antibody against rank ligand based on the key role of this pathway in bone remodeling. So when the first knockouts were generated, it was clear that the pathway was fundamental for bone remodeling, and it's essential for the activation and survival of the osteoclast. And this is very important in the context of bone metastasis. When tumor cells reach the bone, they activate rank signaling for the osteoclasts to kind of make some room in the bone for them to grow. And then this also liberates from the bone different growth factors that feed back on the tumor cells to allow them to grow and survive. So it was clear for my colleagues in Amgen that when they block rank signaling, then you kind of interfere with this vicious cycle. And it's an important or a very good treatment for bone metastasis and also for other bone-related pathologies. So the efforts of this group make possible the approval of denosumab in 2010, which it was mainly kind of 20 years after rank was cloned. So I think that seems like a long time, but it is probably not that much to kind of get a drug in the market. And so when I arrived in 2003, there was many tools for studying rank signaling, and as a postdoc, I got to choose which project I wanted to work at. So in the first months, you rotate with the different groups and you get to know different projects. And I thought that this would be a great one because there were many tools. And there were some findings by the group of Penninger showing the relevance of the pathway in the mammary gland. So I knew nothing about mammary gland. Nobody in Seattle knew anything about mammary gland, but I thought it was cool because most of the development occurs after birth. So it allows you much easier to study the development of this tissue and their different hormones. And it recapitulates proliferation, apoptosis, inflammation. So when the time of after pregnancy or during pregnancy, the main purpose of the mammary gland is to produce milk, which gets at lactation. But then when lactation is over, the tissue gets remodeled and goes through involution to a stage that is very similar to the one before pregnancy, getting ready for a second pregnancy. So I attended a few meetings, and I really like the community of the mammary gland and decided to stay there. So there are two different compartments in the mammary gland, the luminal and the basal compartment, that they are developed through common progenitors in the embryo, but then in the adulthood, they are unipotent progenitors that contribute to the luminal and to the basal lineage. And it was known that progesterone-regulated Rank ligand that is expressed in the progesterone receptor positive cells, and this acts as a paracrine mediator towards the luminal and the basal compartment. So as I mentioned, this paper by the group of Joseph Penninger was published when I arrived, and they showed that the knockout mice, on top of the multiple bone phenotypes, also have a defect in the mammary gland that led to lactation failure. So coming from Maria's lab, where we generated several mouse models, I went ahead and did this gain of function mouse model, the MMTV rank mice, and to everybody's surprise, these mice, despite having increased proliferation and survival of the mammary epithelial cells in contrast of the knockouts, both of them end up showing lactation failure. So no matter that you have no rank or a lot of rank, that's interferes with mammary gland development, and a tight regulation of the pathway is required. And so it's clear that rank plays an important role in mammary gland differentiation. And then again, through the influence of the CNB and Maria, where we use carcinogenesis protocol to study the telomerase mutants, I also look for a carcinogenesis protocol to see if the mice that have generated have an increased susceptibility to breast cancer. And that was actually the case. They got tumors faster and more tumors. And what was even more relevant was that if we inhibit rank signaling through rank FC, we were basically able to prevent mammary tumor development almost completely. So this was very important because demonstrated that rank ligand was the main mediator of the tumorigenic role of progesterone in the mammary gland, and this opened the possibility of using rank ligand inhibitors, the denosumab that was already in the market for breast cancer prevention. So, in 2008, when my postdoctoral period finished, the crisis had already hit the US. So it was a little bit more difficult to kind of stay there. And at the same time, I knew from Puri Muñoz, from Maria's lab, that Manel Estelle was starting a new department in Barcelona, and that he was recruiting PI. So I sent my CV, and all of a sudden, I transferred from being a postdoc in Bill Dugal's group at Amgen to become a PI at the PVC in ECO at the time, and then at EVBEL. So it was a very good opportunity to become a PI. I didn't thought at the time I was gonna make it so quick, but suddenly I was there, in this transfer, kind of alone, and realizing that, all these years of training of the university and the PhD and your postdoc, that you get a lot of skills were not the right ones that you needed to actually lead your team. No? So as a PI, you need other skills that you are not really trained for, related with, people management, money management, a lot of decision making. So it's a tough transition. The first years can be complicated, at least they were for me. And but I got to enjoy it, and it's true that you get to do this job, which is not really a job; it's part of your passion. And the best part of the job by far is the interaction with other people. So people to me is the most important, the PhD students, the postdoc, your collaborators, being able to talk to other people that is excited about the same findings that you are, that, out of this world, you kind of, not many other people understand why you are so excited from this result. You get to pursue your ideas and to see if they are relevant. It's never boring, you're always learning. But it's true that it's tough, because you once you become a PI, you spend less and less time doing science and more time doing administrative work and kind of paperwork, bureaucracy, most of the time looking for money to be able to do what you want to do, and then justifying what you've done with the money. And it's a competitive world, and you really have to be self-motivated, because there will be some ups and downs. But I think, yes, to me, it's worth it. I think it's a luxury to be able to work in something that you love. And it's true that with this great power comes great responsibility, and the lab. Being able to get the funds to train, to hire more people, and to be able to do more science, and how the lab has been growing during these years, thanks to the funding agencies that trust on our work and allow us to grow as a team. This is a big responsibility and a big challenge to lead a big team of people. And as I said, science is a teamwork. So I'm super grateful to everybody that has worked in the laboratory with me during these years, all the PhD students that have worked super hard, and they've made the science that we do, and everybody that contributed, the postdocs, the technicians. So thank you to everyone. And I'm gonna tell a little bit on the work that we do, what we have done during these years. And as I said, once I started working on the mammary gland, I really love the field and the community, so I kind of stay there. This is more of my focus, and we've been always trying to understand the normal physiology of the gland to be able to understand cancer, because it's much easier to understand the tissue in homeostasis that once you go into cancer. Most of the work has been focused on rank signaling, and there's still more and more things that we do not understand. It's true that we've done some other work outside of rank, trying to address mechanisms of resistance to breast cancer therapy, mainly chemotherapy. And as Anna said, understanding the role in the tumor immune crosstalk, which started as a collaboration with Lourdes Planelles that many of you know at the CNB. So we work mainly with mouse models, but trying to make the findings to be translated into patients. So one of the first things that we discovered in the lab, this was one of the first publications where we show that rank overexpression in human cells in MCF10As induce epithelial to mesenchymal transition, and this will also result in the acquisition of stemness features for the mammary epithelial cells, which in the context of cancer, such as BRCA1 mutants, will lead to faster tumor growth and more metastases. So this was true not only in human cells, but also in the mice. So with the mouse models, we show that this rank overexpression lead to increased stemness in the basal and in the luminal compartment. And in the context of mammary gland development, we also discovered a dual role for rank signaling during development. So it acts as a downstream mediator, a positive mediator of progesterone, allowing alveologenesis, but then it interferes with prolactin signaling and therefore with lactogenesis. And this finding that we published several years after the MMTV rank mice, the initial finding that explain why the knockouts and the gain of function and the MMTV ranks have the same phenotype of lactation failure. It's because of this dual role of rank signaling. And in the context of cancer, with other genetically modified mouse models that are oncogene driven and therefore develop spontaneous tumors, if you interfere either pharmacologically or genetically with the pathway, there is a delay in tumor formation, but also less tumors and less lung metastasis. So these were the first findings that suggest that inhibiting rank signaling could have an effect on metastasis beyond the bone metastasis and the bone-related effects, supporting the use of these inhibitors for prevent mammary tumorigenesis, which in fact has led to prevention trials with denosumab in BRCA1 carriers. So, considering this, it was a surprise, and these are recent work on the laboratory where we wanted to foster tumor growth in the polyoma and the NEO models that we had in the lab. So we crossed them with the rank overexpressing mice that were prone to tumorigenesis, and to our surprise, not only we didn't accelerate the tumors, but we actually delayed tumor development in the both oncogene-driven models. And we were puzzled with these results for a while until you kind of take advantage of everything that you learn. And during my PhD, the group of Manolo Serrano was working on senescence. And suddenly we decided to look at senescence, and I kind of reconnect with some of the colleagues at the 413 and decided to look at beta-gal with Manolo Collado, advice for how to do this properly. And we saw that actually there was more senescence in these double transgenic mammary glands, and that indeed only rank by itself was enough to induce senescence in the mammary epithelia, both in vivo and in vitro. And this senescence was through P16P19. So in collaboration with Manolo Serrano, we saw that in the absence of P16P19, rank was unable to induce senescence. And we also show that these rank-driven senescent cells were very sensitive to the BCL2 inhibitor navitoclax. And we could eliminate the senescent cells. And if we eliminate the senescent cells, we also diminish the increased stemness that we have shown that the rank overexpressing models had. So this senescence was fundamental for this increase in stemness that we had described. So in the context of cancer, despite the tumors of the double transgenics took longer to arise, they actually grew faster. And these double mutants had more metastases, and these metastases were enriched in tumor cells that were bipotent for the basal and luminal progenitors. And indeed, if we eliminated the senescent cells with navitoclax, we could attenuate tumor growth and decrease these double mutant cells. So these kind of counterintuitive results at the beginning was led to this finding that rank acting a bit of an oncogene could induce senescence, which at the beginning will prevent or will delay tumor onset. But then once the tumors are there, the senescent cells will foster tumor growth. So this was kind of connecting with the 413 work. And the efforts of the lab during the last years have been focused on understanding if there is also a therapeutic benefit of inhibiting rank signaling for breast cancer treatment. We know that we can prevent, help to prevent tumors, but can we treat them once the patients have breast cancer? So using the mouse models again, we show that if we implanted rank positive and rank negative tumor cells into wild type mice, the cells that do not express rank so decreased ability to give rise to novel tumors and to novel metastases. So we could do this genetically, but also pharmacologically. So if we inhibit with frank ligand inhibitors in the neoadjuvant setting, we also diminish the ability of these tumor cells to give rise to novel tumors. So this will have influence and importance in the clinical setting because as you know, breast cancer patients do not die because of the primary tumor, they die because of recurrence and metastases. So this is what we really want to target. And we show that if we inhibit the pathway, we induce tumor cell differentiation. So this was very important at the tumor side. But then we also realized that RANK was not only expressed on the tumor cells, it's also expressed in the macrophages that invade the tumors, whereas the ligand is expressed in the infiltrating lymphocytes. And being a TNF family member, it's plausible that the pathway plays a role in the immune regulation. So in this crosstalk between the tumor cells and the myeloid cells. So going a little bit more in depth into this story, we analyze the infiltrate of the RANK positive and the RANK negative tumors, and tumors that do not have RANK show an increased infiltration by immune cells, being these mainly T cells and CD8 positive T cells, which are functional because if we deplete the CD8 positive T cells, we rescue this delay in tumor onset that the tumors that do not have RANK show. And we also demonstrated that neutrophils were fundamental for this, and they were, orchestrating this CD8 response. So if we now deplete the neutrophils, tumors, wild-type tumors with expressing RANK now take longer to grow. So these neutrophils were fundamental, and what we show was that RANK signaling will activate in NF-kappa B, will increase this pro-inflammatory atmosphere in the tumor environment that was good for the macrophages and the neutrophils that will then restrict the T cells. So this increase in immune infiltration suggested that inhibiting RANK signaling could also help to or increase the benefits of immunotherapies, and we show this in the mouse models. But what was more exciting, we were very excited with these results, and we wanted to see if this was true in patients. So as Anna mentioned, we had an ongoing clinical trial with breast cancer patients and denosumab, but at the time we were still recruiting patients, so we were not able to look at the results yet. But I knew that Christos Sotiriou at the Jules Bourdet was running a very similar trial, DBeyond, with premenopausal breast cancer patients in early breast cancer, that they will receive 2 doses of denosumab after diagnosis, and then they will collect the samples in the biopsy and the surgery. So when I went to Brussels, told to Christos, and then we shared the results, and indeed, he was observing in the patients the same that we did in the mouse model. So after these two doses of denosumab, there was this increase in the tumor in infiltrating lymphocytes that were mainly CD8 positive T cells. And we saw that actually we could even predict at baseline that there was these patients that will show this immunomodulatory effect will be the ones that have increased RANK signaling in the primary tumor. So this was super exciting to have the preclinical results that you got in the mouse models having the same in the patients. And this suggests that denosumab could be used as an immunotherapy, particularly in luminal breast cancer, where there is very, very little success of immunotherapies. However, despite these evidences, it is true that there is still a controversy about the benefits of denosumab in breast cancer treatment. So there are 2 big clinical trials in the adjuvant setting with denosumab. In the Austrian trial, they actually observe an increased benefit adding adjuvant denosumab to the therapy of care, endocrine therapies in postmenopausal patients with early breast cancer. However, in the CARE, which is a big trial, but including both pre and postmenopausal patients, it's true that they have a slightly higher risk of breast cancer, but they did not observe benefits. So this highlights the need of selecting biomarkers of response to the denosumab in breast cancer, that we are doing an effort to try to identify these putative biomarkers. So as I mentioned before, we have analyzed multiple collections of breast cancer patients with, probably now we have analyzed more than 2,000 patients to show that RANK is expressed in the tumor and in the stroma, unlike RANK ligand, which is rarely found in the tumor cells. And we saw that it is associated with ER-negative tumors. And, there is no association with AIDS. And then, however, so we focus in this distinction between ER-positive and ER-negative because the biology of the disease is very different. And if we look in particular to the ER-negative patients, we saw that high levels of RANK expression on the tumor cells, this is protein expression, leads to poor survival in these patients. So this is very important given the high heterogeneity of the ER-negative breast cancer. And we have some experiments in PDX models showing the increased functionality of the pathway in these triple-negative breast cancer tumors. And some recent findings that we are now actually working on show that there is also an improved benefit, an improved poor survival on the patients that express, on the tumors that express RANK if the patients are postmenopausal. That suggests that maybe the denosumab will be more beneficial in these postmenopausal patients, and we are trying to understand why. It is also important because in our ongoing biomarker trial, we have both premenopausal and postmenopausal patients that hopefully will allow us to answer properly this question. The trial has the same design as DB-JOHN, but it also contains a control arm. So we are currently analyzing the results with the DB-OMAR core. So I had the chance of supervise the PhD of an oncologist, Andrea Betancourt, which is very, very nice experience, and to collaborate with the clinicians and IDVL on this trial and at the ECO. So this is being very, very nice. So I'm not sure if I have time for like 3 minutes.
It's 1 minute already. So
okay, so just going very fast on this part that is not RANK related, just in case anyone is interested, we have generated a big collection of breast cancer patient-derived orthoxenographs, and we've been doing some efforts to try to understand how this particularly triple-negative breast cancer, which is initially very responsive to chemotherapy, to taxanes in this case, become resistant with continuous treatment. So we have done multiple omics approaches, and it's true that our findings suggest this kind of scenario of the great heterogeneity, intratumor heterogeneity, and the clonal diversity in these tumors, showing that after docetaxel treatment, you get an increase in the resistant clone, but then you can interrupt the treatment with these drug holidays and then go back to a stage where the tumors are resensitized to docetaxel. So we found the relevance of CD49F positive cells and also on an amplification in the chromosome 12p arm that suggests to us that we found that sequential treatment with docetaxel and carboplatin could be the key for these treatments. And this is basically all. As Anna mentioned, in 2019, I decided to move again and restart my laboratory at the CNAO. So now I'm closer, very close by. I want to thank all the people in CENIO that has been no, the PhD students, the staff and technicians that are really moving all these projects forward, restarting the group with a pandemic, and also with the clinical part at the IDIVER. So this is our inspiration in the lab to infinity and beyond. And my last slide is for all the, that probably all have seen at the CNB, because they prepared this for Carlos a few years for his anniversary, but these are all, not all, because probably now there are more PhD trained at the DIO, that all the PhDs along the years that have trained. So the CNB has been instrumental to bring in many new scientists to the world. So thank you all.
Well, thank you very much, Eva, for this fantastic talk. About your work and your career. It's nice to see in a picture all the PhD students. Well, in the past, now there are many.
There are more now, probably. Yeah.
Yeah, many more. So because there is a delay with the YouTube, whilst we are waiting for questions to arrive, I have a couple of curiosity. One very quick one. One is, why did you think the denosumab works much better in post-menopause women? Do you have any idea?
So this was kind of unexpected, because in reality, everybody, because rank ligand is downstream of progesterone, everybody, me included, thought that it was going to be more efficient in premenopausal breast cancer. We are trying to understand this. This is current ongoing in the lab, but it's true that in one of our hypotheses is that, as happens in the bone, so after the estrogens drop, after menopause, there is a decrease in OPG, which is the inhibitor of the pathway, and there is increased rank signaling in the bone. This is what leads to osteoporosis. So we think that in cancer, we may also have this increased activation of rank signaling in these postmenopausal tumors. So one thing is the expression, and the other thing is the activation of the pathway. Mm-hmm. But, as I say, there is all this more pro-inflammatory environment in postmenopausal. There are some metabolic changes. So, we are currently working on this, but these are some of the hints that we have.
Okay, and regarding this inflammation you mentioned, rank has a role in other type of cancer, more inflammation driven, like colon cancer or other type.
So we don't know about colon. So this was one of the things that I started to explore during the ARC, because rank is expressed at very high levels in the intestine, very high. But when we knock it out, there is not a major phenotype on the epithelia. So what it's known for rank, and it was published kind of concomitantly as we were generating the models, is that it's important for the establishment of the M cells and the peripheral tolerance in the intestine. So that is the phenotype of these mice. They do not have M cells. That change the microbiota composition, but we still don't know what it may be doing in the epithelia. There has been some publications demonstrating the relevance of rank in lung cancer, which is clear, I will say. And also, I think it's an important role in many leukemias, lymphomas, and so on that needs to be further explored.
Okay. Well, I'm going to read you some comments in the YouTube. Teka Galbao say, "Oh my God, great to see the photos of Christmas party and canoeing." And also Domingo Barber say, "Excellent talk, Eva. Thank you very much for participating. You have explained very well the different phases of the scientific career in addition to your excellent work, inspiring for the youngest researcher." So I joined Domingo's words because, truly has been inspiring your talk. Okay, well, thank you very much, Eva.
Thank you very much, Anna. And I hope to see you all in person soon. Now I'm very close, so I can stop by easy.
Okay, bye.
Thank you very much, Eva. Great talk. It's amazing, your scientific trip around the world and around different cities in Spain. That's fantastic. And the talk was really, really impressive. And I want also to thank especially that you remember our times initiating the department. That's a great time for all of us.
It was amazing, yeah.
Sure, absolutely. And then we are going to finish the last talk of the meeting is correspond to the Department of System Biology. Then Victor, can you start when you
Okay, well, good afternoon, ladies and gentlemen. It's a pleasure being here and introducing the speaker that has been picked up by our department. And his name is Pablo Nickel. So Pablo holds a position of senior researcher and group leader in the Center for Biosustainability of the Danish Technical University in Lyngby. That is a place close to Copenhagen. So before I start talking about Pablo, you know, when I was thinking on what could I say, it came through my mind that is kind of a common saying in our community, this idea that if I could look farther was because I sat on the shoulders of giants. And this is one of the things that we used to say. Well, I tell you, my experience is basically the contrary. So if I could look farther, is thanks to the fact that wonderful collaborators landed in my laboratory, and they were able to show me perspectives that I would have been otherwise unable to identify by myself. And one of the best representatives of this situation has been Pablo. So Pablo comes from Argentina originally, and he made his studies in the University of Buenos Aires and the University San Martin, also in Buenos Aires. And then he finished his degree in biotechnology in the year 2004. So after that, he started a PhD with a lady called Beatriz Mendez, and he worked for some time during his PhD in production and molecular biology of polyhydroxybutyrate production in E. coli. And for those who are not familiar with the field, this compound is a bioplastic. And so even by that time, that discussions on sustainability and so were not as common as we have today, then Pablo was able to identify a topic that later has developed as something very important for our future endeavors in industrial biotechnology. So by the time he was finishing his PhD, he apparently came across one of my talks in a meeting of the American Society of Microbiology in Boston, if I remember well. And then for whatever reason, he found interesting the work that we were doing with Pseudomonas and metabolic engineering or genetic engineering of bacteria for environmental release and environmental remediation and so. And then 1 year later, he approached me in another meeting we had in Barcelona. And then we started a conversation and we identified common grounds for shaping a proposal for a postdoc. So he applied for an EMBO fellowship and he was granted an EMBO fellowship. He later applied for a Marie Curie fellowship or contract, and he was also granted it. So he was able to combine various, say, grants. And at the end, he remained in my laboratory for 5-6 years. And I have to say that during the period of time that he was in my group, it has been one of the most, say, productive and amazing periods of time that we have been through in my lab in the CNB. So what Pablo, convinced me, and Pablo delivered to the whole group and the whole perspective of our research agenda, was the importance of metabolism. So, we work in an environment of molecular biologists, and let's face it, any idiot can make a PCR and any idiot can clone a gene. But very few people can handle metabolism. Metabolism is not for everyone. You need to have a sense of chemistry, thermodynamics, kind of global view of things and so on. So I think that it was thanks to Pablo that, we were able, for the period of time that he was in my group, to really start focusing in important topics that otherwise, as molecular biologists, we had never paid attention to. So, that's for the time being all I wanted to share with you. So if at the end there are questions, I will get them through our coordinator. And I hope that you consider Pablo's talk as the grand finale of all this wonderful session that we had today for the celebration of the 30th anniversary. So Pablo, the floor or the screen is yours.
Thank you very much, Victor, for the very nice introduction. Let's see if I can share the screen now, because I'm not using my computer. Here we go. So I hope that you can hear and see the presentation. So I will start by thanking Victor for the very nice introduction, and of course, the systems biology program.
Pablo, can you blow up the screen so we don't see it full screen?
I think I did, actually. Let me try again.
In the presentation mode, that's the thing you have to.
Yeah, it was in presentation mode in the computer. Can you see it as such now? Because the thing is that I'm not using my computer, so in my screen it shows up as a full presentation, but I can maybe try again. Okay, so I'll try again. Okay, so meanwhile I will just try to figure out. Yes, so let's see if I can do it now. Okay, this should be presentation mode now, I assume.
No, it's not. You have to go to, I mean, for us, it's just the PowerPoint.
Okay, then instructions.
Let me see, I'm going downstairs to see if I can help.
Meanwhile, I can start talking. So I am fully aware that the only thing that is keeping us from the paella is my talk. So I promise that I will try to deliver a dynamic and focused talk. By the way, I'm sitting in a place where I can see the paella being done. So I feel the pressure, just visually in front of me. So I will start by saying that I am very, very glad to be here, even when this is kind of a rather peculiar format for me. So we'll see how it goes. And of course, we have to adapt to these little bumps on the road. But I have Susana here, so if that works, because as you can see, I'm sharing the screen and it's in presentation mode. So
yes, it is in presentation mode.
Mm-hmm.
So can you go back to stop sharing?
Absolutely, yes.
And then, because when you share, there are two options. If you click in here, and instead of showing that, so
This one? Okay.
Let's see.
So is that now in presentation mode?
Now it works.
Thank you.
It is fast before I go.
It doesn't, but I think I can use this maybe.
Yeah.
Good, very good. So that being said, thank you very much for the help on that. The little bumps on the road that one has to face in this formats, right? But here we are. So the idea of the talk today would be to focus a little bit on a couple of things that shaped the way that I see science, and that formed the basis for the research team that I started in DTU Biosustain. So I will try to walk you through some of the ideas that were concocted here in CNB, and then they take the shape of the things, or many of the things that we are doing now in my research group in Denmark. So as Victor said, I am originally from Argentina, and when I landed this position here in CNB, I started as an EMBO fellow for 2 years, and then that transformed into a Marie Curie fellowship, where I further developed some of the ideas that were started in the EMBO fellowship. So I was able to retrieve a couple of presentations that we were discussing in the context of lab retreats while I was around. And I got this one. So this was my very first presentation in a retreat in 2011 in Trujillo. And in there you can see what the original EMBO application was about. So as you can see, the idea was to refactor Pseudomonas putida at the genome level to legalize metabolic properties, to bring about new to nature functions in Pseudomonas putida. I was very much attracted by that prospect because as Victor mentioned, I had the opportunity to listen to one of his talks. The very first one that I had the opportunity to listen to was in in Boston in 2008 in the context of the ASM meeting. And even when it was mostly focused on genetic engineering of Pseudomonas, it was very clear by then that Pseudomonas has got a lot of metabolic properties that were worth pursuing. And one of the things that I had to mention is that back then I was working with E. coli only, right? And within the bacterial realm, gram-negative, granted, but still I wasn't exposed to the power or the potential of Pseudomonas putida. And that was something that caught my attention back then. And as Victor mentioned, 1 year later, we applied to the EMBO fellowship, where the idea was essentially doing what is in here. And even back then, what we did was to think about a couple of experiments that could be implemented to the sake of studying metabolism in Pseudomonas. And some of the experiments we did back then, some of my very first experiments or experiences with Pseudomonas were rather simple. Even plate experiments, just, you know, exposing cells to trace agents and see how they behaved. And this actually shaped one of the research lines that I was working with here in CNB, that was essentially studying how glycolysis is regulated in Pseudomonas. And just to cut the very long story short, one of the things that we discovered in collaboration with colleagues here and in other places as well, is that there is a very particular architecture of glycolysis and catabolism in general in Pseudomonas putida, something that again shaped a little bit the ideas that we will discuss later on. So on that, you know, I also got this one. This was my second retreat, where you can see that the portfolio of projects that I was running in collaboration, either alone or in collaboration with other colleagues in the lab, further expanded into other things. And then this was my last retreat around here, that it was in 2016 in the Monasterio de Piedra. And that was a really fantastic and exciting retreat. So by then, the portfolio of projects that I was involved at further expanded into this, as you can see here. And this is a table that, you know, if there is a piece of advice that I will give to anyone starting their own group, is to have a table with projects that looks like this. This is an idea that Victor promoted in the team back then, and I think it's still around, and it's something that I totally copied, because I think it's a very, very natural way to picture how developed the projects are or aren't. As you can see in here, you know, there is a column for embryonic and wishful thinking projects. Some of them were embryonic in 2016. Some of them are still embryonic as of 2022. But I want to draw your attention to the very last line here, 31st in the row, that says harsh biochemistries in Pseudomonas putida. This is what I did later on is about. Establishing biochemistries in Pseudomonas putida as a host that will be virtually impossible to establish in any other chassis, in any other platform for bioproduction. And the idea here resonates a bit with what we call synthetic metabolism. And let me walk you through very briefly about what we understand as synthetic metabolism in bacteria. The whole thing here is that, as I mentioned before, building on these ideas of very special metabolic architectures in bacteria, we started to think back then whether the extant biochemistry, the set of bioreactions that we see inside the cell, that we can measure, that we can see as active reactions within the cell, are the very limit of all the reactions that are possible within a given cell. And it's very obvious that the answer is no, but then the question is like, how far can we go into engineering the metabolism of bacteria, and in this case Pseudomonas for obvious reasons and reasons that we will discuss in a second, how far we can push the extant metabolism to adopt new reactions. And in here you can see kind of two realms, right? One is the realm of the chemistry that exists in the cells, but another one is the realm of neobiochemistries, is the realm of reactions that could happen inside the cell, potentially, that also use non-biological atoms as part of the chemistry they run. And that was essentially the seed for some of the projects that we are running in Denmark right now. And this synthetic metabolism is just a framework that we adopt to push the extant biochemistry to the very boundary of new to nature bioproduction. And we try to blend atoms that, as I mentioned before, are usually not part of the biological agenda. So we are particularly interested in establishing biohalogenation in Pseudomonas putida, but also assimilation of compounds that are not part of the palette, say the biochemical palette of bacteria. So as you can see in here, this framework of synthetic metabolism is split into biosynthesis, synthetic metabolism for biosynthesis, for production, and synthetic metabolism for assimilation. With the core concept of harsh biochemistries, reactions that will be difficult to handle, that are usually part of the chemical realm. But the question here is, how many of these reactions, of these metabolisms, can be biologicized, if the term is correct, right? So how many of those reactions can we bring into living cells? And the term, can be further expanded. This is actually very cool for discussion, I think, because it's just a concept, right? It's not something that we are doing, but it's something that I hope to see in the future, which is, instead of recoding nucleic acids as we are used to as of today, can we actually recode biochemistry, the very building blocks that make up full life, right? So we are used of biochemistry that is based on carbon, hydrogen, oxygen, nitrogen, and so on. The question here is like, can we bring completely alien atoms and recode the core biochemistry of cells using them? And this is a little bit what the whole thing is about. So this is the Center for Biosustainability. That picture was taken on a particularly sunny day, but we are sitting in the Copenhagen region in Denmark. And essentially, this is one of the 4 centers, the so-called centers for excellence of the Novo Nordisk Foundation. And the Center for Biosustainability, as the name implies, has as a major role to provide greener solutions to the bioproduction of chemicals, drugs, materials. And it's in this context that I brought some ideas of these harsh biochemistries, new biochemistries in bacteria. So this is the team. This is not the most updated picture so far, but it's one of the recent pictures of the team. The name of our group there is the Systems Environmental Microbiology, and as you will see in a second, we are interested in bringing these chemistries and having a systems view of what the effects of recoding metabolism are, right? So we have different axes of interest in terms of major activities that form part of our interests in the Systems Environmental Microbiology group. But today I would like to focus a little bit on synthetic metabolism for production and assimilation, and I'll just give you 2 examples of those 2 cases for further discussion later on. So this is a little summary of the things that I've been saying before. And remember, again, this concept of using synthetic metabolism is just an idea, is a framework whereby we try to implement completely new ways of handling chemicals in bacteria, right? So on one hand, of course, our major interest and that of many other labs around Europe and the world is to provide alternative platforms for bioproduction. And we really think that Pseudomonas is a good case, at least for some of the products that we have in mind. And that means that, of course, we need to construct very complex phenotypes in some cases, bring about new reactions, as I mentioned, that require a deep genome engineering and genetic engineering program of Pseudomonas. And the output of that is establishing synthetic metabolism, right? That, as I mentioned, is essentially under 2 main blocks. One of them is halogenation, and we are particularly interested in fluorine as a new-to-nature atom for recoding biochemistry. And then in the realm of assimilation, we are very much interested in the assimilation of C1 compounds for obvious reasons, but we will discuss that in detail later on. Another thing that I would like to mention is that in connection to one of the things that Victor mentioned during the introduction, I've been working with production of polyhydroxyalkanoates since I was doing my PhD thesis in Buenos Aires. And this is something that I wanted to resurface. But in this case, under the context of whether one can use these polyhydroxyalkanoate production strategies to blend in new atoms as well. And as you can see here in the formulas, that down here in the bottom row, we are interested in a direct path of assimilation of C1 and production of new polymer compounds, and some of them containing fluorine. So let's dig deeper. So just to provide a little bit of a context as of why Pseudomonas, right? I mean, people working with bacteria know very well that Pseudomonas can be found in a number of different environments, and that one of the consequences of the environmental versatility, let's call it that, is essentially the core reason why we adopt Pseudomonas as a chassis. Metabolic versatility, metabolic robustness, a number of reactions, a very rich metabolism that we can play with, right? And this has been the motto, the leitmotif, since I started working here in Victor's lab, and it continues to be exactly the same as of today, where we move into more and more complex biochemistries. But, you know, Pseudomonas is an excellent starting point for that matter because of the versatile metabolism, core metabolism that it has. And of course, it continues to be our favorite pet, so we do all sorts of very weird manipulations with Pseudomonas that I hope to give you some examples about very soon. Our approach to ingrain these synthetic metabolisms in a non-traditional host, and when I'm saying non-traditional host, I'm referring to Pseudomonas in the context that it is being used for bioproduction, it is being used as a laboratory host for sure, and it has been for decades now, but still is not at the level that we can compare the ease of manipulations and the type of manipulations that we can do to the level of, say, E. coli or Saccharomyces, right? There are a lot of effort in that realm, and several labs, not only us, but several labs around the world, several labs here in CNB are working towards the same goal. But the approach here is essentially to adopt the DBTL cycle, the design, build, test, learn cycle of synthetic biology, and, you know, drop in Pseudomonas putida and try to get the synthetic metabolisms in, produce these new to nature compounds, and understand, as I mentioned before, from a systems level perspective, omics perspective, what are the consequences of those manipulations, and learn to essentially implement a new set of modifications in the next cycle. So this is a little bit the same concept, but it's something that we are very much interested in is in automation. So some of these logs within the DBTL cycle are very time consuming. Some of them are not automated at all, they're almost artisanal in nature. So something that we rely on and we very actively work towards is the automation of some of these activities. And one of them is multi-omic characterization of Pseudomonas, that we really think it will ease the design of synthetic metabolism. So let's see what happens inside the cell, right? So I mentioned before these extant biochemistries. So from a helicopter view, of course, the core metabolism of Pseudomonas and many other organisms as well, but Pseudomonas is remarkable in the sense that this funnel is pretty broad, but essentially you can picture it as it is in here. So you have a core metabolism, and you have, like, you know, this bow tie sort of structure where you have to the left a very broad funnel of substrates that can be incorporated and processed by central metabolism into products of interest. And this is really very broad because this is the versatility of central metabolism in Pseudomonas, as I mentioned before, is the reason why we use it for bioproduction. The question here is, can we expand new entry points into this metabolic funnel, into this metabolic bow tie, right? And we are essentially interested in two things as examples. One of them is whether we can introduce fluorinated building blocks as a starting point for bioproduction, and whether we can use C1 feedstock as carbon substrates for Pseudomonas bioprocesses. So just to summarize a little bit the type of activities that we do, as I mentioned before, we adopt this DBTL cycle narrative where we have design, build, test, and learn processes. And in the build, of course, we have accelerated genome engineering tools. As I said, most of the manipulations that we do require to introduce a number of mutations, not only one or two, but a number of very radical genome engineering approaches. So we are very much interested into this part. The second part is how to convince the cells to run these reactions, because these harsh biochemistries have many consequences. Some of them, involve stressful or toxic intermediate substrates. So growth coupling, convincing the cells that they have to run synthetic metabolism in order to survive, is a strategy on how to drive evolution of these pathways. Then in the learn and redesign cycle, we have the core activities of metabolic engineering. And in here, I'm just showing you an example that is later, I'll refer to this later on, but this, I think, captures very nicely the power of Pseudomonas fluida, which is like, the inherent, the native machinery for consumption of aromatic substrates is something that we have refactored and built upon for production of fluorinated building blocks. And this is something that is almost unique to Pseudomonas, other species as well, but it's not as widespread, and yet another reason why we use. So just one example of this block of synthetic metabolism for biosynthesis. As I mentioned before, we are very much interested in bringing fluorine to life. Why fluorine? So fluorine is an absolutely amazing element. It would require a completely different lecture and just one lecture devoted to the properties of the C to F bond, the carbon-fluorine bond, because it's absolutely amazing. Suffice it to say that it's the most electronegative element in the periodic table. It's very abundant. It's really very abundant in the Earth crust, but it's very reactive. So you never found it as elemental fluorine, which is what we have pictured here in the upper part in the cube. That's liquid fluorine in the elemental state. But we are very much used to the presence of fluorine. We actually brush our teeth every morning with toothpaste that contains fluorine or fluoride salts. And the reason for that is essentially the chemistry behind fluorine that we will not enter into much detail, but this is an absolutely fascinating aspect. But what happens when fluorine is combined to carbon, when it forms C2F bonds, is that the number of compounds and materials that can be produced, organofluorine materials, really expands a lot the possibilities of our daily life applications. We are very much used to cooking over Teflon, and Teflon is no other thing than a polymer containing a lot of fluorine. What is so important? Why is fluorination so important? Well, fluorination of carbon chains brings about the fluorine effect, the so-called fluorine effect, that is imparting hydrophobic and hydrophilic properties at the same time. And this is absolutely unique to fluorine. I mean, that's the reason why we cook on Teflon. It repels water, but it repels oils as well. This is a unique property of the C2F bond. Other applications as well, surfactant clothing, that is very important if you live in Denmark, believe me, or for the clothings for outdoor activities. But most importantly, the C2F bond is widely present in the pharma industry. And that's one of the reasons we are very much interested in the production of drugs containing fluorine, because 1/4 of all the pharma drugs contain fluorine one way or the other. And most importantly, we don't have absolutely any way to put fluorine on an organic molecule that is bio-based as of today. So we were relying on traditional chemistry, very dark chemistry for fluorination, and we tried to change that. And essentially, our approach to synthetic metabolism, again, I don't have time to go into details, but I just want to give you two examples of bottom-up and top-down fluorination. So essentially, the bottom-up approach is how to create a C2F bond inside the cells, whereas the top-down is biotransformation of substrates that already contain fluorine. So for the bottom-up, one thing that one of the postdocs in the team did very nicely is to create a system, a synthetic circuit that is triggered by the presence of mineral fluoride for the production, the in vivo production of fluorinated sugars and nucleotides. And the importance of this is that both fluorosugars and fluoronucleotides are direct building blocks for fluorinated drugs that, as I mentioned before, as of today, we can only obtain chemically. So this provides an alternative to that, which is bio-based. And it's essentially, you know, this is particularly interesting for us because in this case, there is a carbon to fluorine bond formed de novo in vivo. And this is interesting because, as you will see in a second, there is only one enzyme that can do the job, okay? And in the top-down biofluorination approach, as I mentioned before, we have biotransformation. And this is related to the properties of Pseudomonas in terms of degrading aromatic substrates, because what we do here is rewire the machinery that already exists in the cells and just optimize it such that fluorobenzoic acid, aromatic compounds containing fluorine, can be transformed into building blocks. And this building block that we are particularly interested in is fluoromuconate. Muconate is a platform chemical for production of materials. And now what we are trying to do is produce these materials, but with a fluorine derivative on it that brings about physicochemical properties to the material that are new to nature. So we are really exploring what those properties are and whether they can be tuned for what they are called smart wearables. So that means essentially, you know, new to nature properties, physicochemical properties brought about by the presence of fluorine. One thing that I mentioned is that we are particularly excited about the bottom-up approach in the sense that forming a C2F bond is something that in nature is known to be done by only one enzyme that does it very badly. So the so-called fluorinase, which runs the reaction that you can see here, the fluorination, the direct fluorination with fluoride as a nucleophile on some isodenosylmethionine. This is an enzyme that was discovered kind of recently, less than 20 years ago, in an Streptomyces species. But there are only 6 fluorinases out there, or there were 6 fluorinases out there. So I just wanted to highlight the work of another postdoc who was only 1 year in my lab as a postdoc, and now she's a group leader in CIB, Isa Pardo, and she was able to discover the fastest fluorinase in nature. That is still slow, don't get me wrong, but much better than the others that we were working on. And interestingly enough, this fluorinase comes from an archaea. So this idea that fluorinases only evolved in Streptomyces was challenged by this idea, right? So a faster fluorinase with which she constructed a synthetic circuit for production of fluorinated sugars and nucleotides in the same concept that I mentioned before. And with this trick and this new enzyme that was kind of hidden in plain sight in archaea, she was able to produce the highest fluorometabolite concentration reported so far, either in engineered bags or in bags that can handle fluorinated sugars as well. So we were very happy about this. And this is just a part of a let's say, long-term objective that is essentially building up on fluorination and legalizing fluorination, to use the same word as I introduced when I was talking about my EMBO fellowship that got me here into CNB in the first place. And the idea here is to expand the number of fluorinated molecules that we can produce in a pipeline sort of a structure where we can add different parts to the molecule, like chemoenzymatic approaches, and in this modular way, expand the portfolio of fluorinated sugars, nucleotides, and drugs thereof that can be produced using biological systems. So that was it for the synthetic metabolism approach for biosynthesis of new to nature compounds. Now, a quick example on assimilation. The concept is still the same, and the framework, the conceptual framework is still the same, but in here the problem is different. The problem is very easily announced in the sense that we all know that we have a major problem in terms of emission of greenhouse gases. And I took this snapshot. This is the CO2 concentration in the atmosphere as of yesterday, where we can see that the trend is unfortunately upwards and clearly upwards, so there is no discussion about that. And you know, this has resonated in a number of conferences and ongoing discussions. But the question here is like, what do we do with that, right? So we need efficient, smart ways on how to capture CO2 and C1 derivatives thereof. And in this case, what we selected as a model problem was a soluble form of CO2, which is formic acid. So the whole idea here is that CO2 can be converted into formic acid relatively easy by electrochemical conversion using renewable energy. So it's very cheap, especially in places like Denmark, that most of the energy platform is renewable. There is a lot of surplus energy, surplus electricity that can be used for reduction of CO2. And once one has formic acid, one can either use native organisms that can consume C1 substrates or engineer a synthetic metabolism for C1. And Justine, a PhD student in my lab, took that challenge in Pseudomonas putida to design a synthetic formatotrophy. We will not enter into details, but you can see in here that the cycle, or in this case, the bicycle structure of metabolism is a leitmotif of many of the synthetic metabolisms that we work on. And using a number of approaches that rely on bringing an evolving new enzymes for efficient assimilation and attachment of C1 molecules into metabolism and adaptive laboratory evolution, the thing was able to establish synthetic formatotrophy in Pseudomonas putida. To the extent that we can use formic C1 substrate as the only carbon substrate to grow Pseudomonas. Slower than sugars, granted, but C1 assimilation in a direct route. And this is, again, just one module of a longer and more complex type of approach, which is what to do with that. And this is essentially our proposal for using Pseudomonas as a platform for C1 assimilation, whereby formic is the substrate, as you can see in the upper part of the picture. And from there, one can create monomers that, again, can be polymerized to form polyhydroxyalkanoids, and performance materials thereof containing C2, C3, and C4 monomers, right? That is glycolate, lactate, and hydroxybutyrate as building blocks derived from C1 for polymer production in vivo. One thing that I would like to mention that I think is kind of is considered a byproduct, but for us it was a very nice addition, is the fact that once the cells assimilate C1 substrates, they produce the polymers. Polymers get extracted and used for further applications, cosmetic, medical, and packaging, usually. And what you are left with is a lot of residual biomass. So that's bacterial biomass that doesn't contain polymers. So what to do with that? And essentially, one thing that we are in discussions with a company right now is to use the residual biomass as an animal feed. And I don't know whether you know about that. I didn't before moving to Denmark, but Denmark is one of the main, if not the main in Europe, producer of pork meat. So they have pig farms, and this residual biomass is very nutritious, very rich in nutrients, and that can be used as animal feed. And this is something that we are doing. So I will kind of wrap up here. So we have a little bit of time for discussions now, but a little bit of what lies ahead, right? So this whole concept of synthetic metabolism that started back then, talking about harsh biochemistries, now is mature enough to start thinking about the scale, right? So we are playing with older synthetic metabolisms for biosynthesis and assimilation and trying to construct more complex phenotypes. So we are very actively working in cell-free systems as well for production of some very complex molecules that will be just too damaging to produce in vivo systems. So we are using cell-free extracts for those and building up on in vitro cascades for their production. We're doing a lot of enzyme engineering. So the building blocks for synthetic metabolism are new enzymes. So we try to code these new chemistries in new enzymes, and that requires a lot of rational and just natural evolution as well. As I mentioned before, we are at the stage that some of these fermentations can be scaled up. So we are trying to do that, especially for assimilation of C1 compounds, and also letting evolution take a part of this in long-term adaptive laboratory evolution to, as I mentioned before, at least in some cases, couple production with growth. And we are actually interested in real-life applications because the examples that I mentioned before, they are still at the lab level or like, low technical level, right? But we really want to see whether these things make sense from a techno-economical point of view and a life cycle point of view. So this is something that we are doing. With this, I will just conclude by thanking to the real heroes here, because all the work that I presented has been done by this bunch of very talented people who are the group in Denmark, to whom I am totally devoted to, and then the funding support by the European Commission, the Nomonores Foundation, and a number of different interactions with companies here in Europe and United States. Well, some of our collaborators, you can see CSIC down there, so we are still actively collaborating with colleagues here in Madrid. And I would like just to conclude with this picture that was taken a couple of years back, actually, my last year around here, was besides bringing, ideas about harsh biochemistries and synthetic metabolism, I was left with a lot of connections, a lot of friends, of very talented colleagues, the mentorship by Victor that has been instrumental ever since. And with this, I will just thank you very much, you all, for listening, and I'll be very happy to take questions and discuss now and with the paella. Thank you very much.
Okay, thank you. Thank you very much, Pablo. It was a wonderful description of your past and common and ongoing and future interests. So we're slightly late, so we try to shorten up a bit the discussion. Maybe during the paella we can return to that. There's a question that you have not tackled, and in my opinion is important, is the stability of the bags that you develop in the laboratory with these unusual chemistries. I mean, is there any, number one, is there an evidence that they are not very happy with the type of pathways that you introduce? And number two, I mean, if they are unhappy, are there any good ideas of how to handle this long-term performance?
That's a very good question, Victor. So in principle, we haven't found for some of the, for instance, fluorinated building blocks, a major impact on cell physiology. We really haven't. But for some of the shortest organic acids that contain fluorine, they tend to be toxic. So the way there is essentially using these growth coupled selection schemes, whereby you have a molecule that is split into the product of interest, and the other half of the molecule feeds biomass production. So that forces the cells to run the reaction, because one of the, let's say, part of the molecule, the original molecule, goes to biomass production. Cells will not grow until they run that reaction. And that has been very instrumental in establishing synthetic metabolism for biofluorination, because the cells are essentially evolving on the way of, using those reactions to grow, otherwise they wouldn't. So that works very well.
Okay, thank you. Well, I think the time has come to finish this contribution. And Mario, you have the word now.
Yep. Thank you very much, Pablo, for this amazing talk. Thanks that I want to extend to the rest of the speaker. I think that it has been a very brilliant workshop to celebrate our anniversary. And I want to especially thank Susana for helping all of you and all of us to maintain the connection and to develop everything for the success of this meeting. And now Susana said that it's interesting that everybody now in this Zoom meeting, in this Zoom application, maintain your connection and connect your cameras just to take a picture of the group. And then for those that are here, enjoy the paella, and we will see in a few minutes. And for the rest, we wait for you at the CNB. You know that is your home. I mean, you come back whenever you want. Thanks, everybody.